Okay. Good afternoon. Good afternoon, everybody. So it is my great pleasure to welcome everyone to this year's CBR LSI Research Day. My name is Hugh Kim. I am the director of the Center for Blood Research, and we're very, very happy to have everyone here, and we'd like to also welcome everyone who is tuning in remotely through Zoom. I'd like to begin by acknowledging that we are on the traditional, ancestral, and unceded territory of the Musqueam people. And very importantly, I'd like to acknowledge with extreme gratitude the sponsors for this event. And our events could not take place without the generous support of our sponsors. And as well, to acknowledge all of the efforts that are made behind the scenes to put together the event and also to promote the event. So that's great thanks to Mira and Jessica from the CBR office, and to Sharda and Dipali from the LSI office. So if you see any of these individuals, please be sure to reinforce our great thanks for their efforts. So the annual research day is a terrific event because it's not just the great science, but it's also the informal setting, and it's really one of the flagship events that we have here at the CBR every year. So we're really, really happy. We look forward to it every year. So we have a great program lined up today. So we have the focus of the CBR Research Day and the LSI Research Day is to really highlight and acknowledge the hard work done by our summer students and all of the individuals who spend the effort to guide them and mentor them in the labs. So they'll be summarizing in 3 minutes, and no more than 3 minutes each, all of their hard work for the past 4 months. So we'll start with that. And we have our annual tradition of awarding the Neil Mackenzie Mentorship Award for Excellence in Mentoring, followed by a terrific keynote address from Dr. Bandari, who's joining us from Hamilton, Ontario today. And he literally just flew in this morning. So we're very, very happy to have Dr. Bandari with us today. And followed by, again, the posters and the poster judging session that happens. And that's where for any conference, that's where a lot of the free-flowing discussion happens, and it's really a big highlight of the event as well. So please do take the time to look at this QR code. We have a People's Choice Award where everyone can vote on their favorite oral presentation. The voting will close at 3:30 p.m., so please do participate in this. So now for the main event. So we have our students who will be summarizing their summer's efforts in 3 minutes or less. So just a reminder to our student presenters, okay, to look forward to the audience, not back. And the target is 3 minutes, and we will be enforcing a very strict 3-minute rule. And instead of the hook, we have, keeping with tradition, Kevin, hit it. That will be the reminder that their time is up if you hear that. So please, so thank you for respecting that. So let's get started with that. So we'll invite our first presenter. Okay.
Okay. Hi, I'm Alice from the Tropini Lab, and I'm excited to introduce you to my project, which is to genetically engineer the probiotic E. coli Nissl 1917 to report on osmotic and oxidative stress in the gut. So our gut hosts a wide range of physiochemical properties, including fluctuations in pH, temperature, oxygenation, as well as osmolality and oxidative stress. Osmolality is very similar to osmolarity, but we don't care what the specific solute is. And osmotic stress can occur due to, for example, malabsorption during celiac disease. Oxidative stress often happens in our gut due to inflammation and immune cell activation. The bacteria that live in our guts have endogenous mechanisms to be able to sense and respond to these changes, and my goal was to leverage this to produce a fluorescent signal when these changes occur. The first step I took to do this was through transcriptomics, so I wanted to identify genes that are specifically differentially expressed in each stress condition, but not the other. So I had E. coli Nissl grown in baseline, as well as 4 different osmolytes, including salt and polyethylene glycol, or PEG, a common osmotic laxative, as well as in lower pH, oxidative stress with hydrogen peroxide, as well as increased temperature. And like I had hoped, I found genes that were specifically upregulated in each stress condition, as shown in the orthogonality plot here. The next step was to amplify the promoter regions from the genes that I had found, and I got the promoters to drive a fluorescent protein, SFGFP, and the goal was to hopefully see an incremental increase in fluorescent output as the stressor concentration increases. So I had 26 osmolality candidates, as well as 19 oxidative stress promoter candidates. From the 26 osmotic stress candidates, 7 were able to drive fluorescence under salt and PEG, and out of these 7, the promoter from the Pro-V operon had the best response, with a 5-fold increase in salt and a 2.5-fold increase in PEG. And by fold increase, I'm talking about how many times greater the fluorescent output is at that osmolality compared to baseline. Out of the 19 oxidative stress candidates, there was 4 promoters that were able to drive fluorescence in hydrogen peroxide. And out of all of these, HPC had the best response with a nearly 4-fold increase in 800 micromolars of hydrogen peroxide. So I had these 2 best promoter candidates, and I went on to do some more fine-tuning, including a synthetic RBS and antisense transcription. I would love to talk to you more about this at my poster. It's poster number 1. And I'm currently working on integrating my double reporter construct into the chromosomal DNA to hopefully test in vivo really soon. Thank you.
Hi, everyone. My name is Aaliyah, and this summer I researched the early inflammatory and aging signatures induced by miR-146A knockout in healthy hematopoietic stem cells, or HSCs, with Dr. Christina May, Josh Bridgers, and Vivian Wu. miR-146A is a microRNA. MicroRNAs are non-coding RNA molecules responsible for mRNA degradation and translational repression. Our lab is interested in the pathogenesis of myeloid malignancies, such as acute myeloid leukemia, or AML. AML is a deadly disease with a survival rate of only 10% in individuals over 60. We know that several pro-inflammatory cytokines are increased in patients with AML, and our previous data has shown that depletion of miR-146A with aging leads to inflammation, which potentially leads to altered HSC functions. Our lab has previously shown in both miR-146A knockout mice and in AML patients with low miR-146A levels upregulation of key inflammatory pathways TNF-alpha and IL-6. We also see upregulation of an aging signature. Together, this gives us inflammaging of HSCs, potentially contributing to myeloid malignancies. However, we don't have data on normal human HSCs. The question we are asking is whether targeted knockout of miR-146A can affect inflammatory signaling and aging pathways in young human HSCs. To knock out miR-146A, we took cord blood cells and used CRISPR-Cas9 to generate 2 individual knockouts. Here we can see from the digital PCR results that the guides can achieve a very efficient knockout of about 85 to 95%. I then analyzed the RNA-seq data and showed upregulation of TNF-alpha and IL-6 as expected, as well as upregulation of the aging signature. However, it was a surprise to see that the miR-146A target genes were not differentially expressed. This is unexpected because if miR-146A is down, theoretically, its direct downstream target genes should be up. This raises the question, why did these results not completely recapitulate the previous human AML dataset and mouse model? One hypothesis is that while miR-146A was knocked out, the seed sequence was not fully disrupted. The seed sequence is crucial for target gene recognition. Based on where the Cas9 enzyme cut, I found that the predicted structures of the knockouts do retain the seed sequence and thus could retain some miR-146A function. The current goal is to pursue new methods to fully disrupt the miR-146A gene. Another thought is that since this experiment was performed in cord blood, a young source of cells can have a different inflammatory effect than adult cells. Therefore, this experiment should be repeated with adult bone marrow cells. Thank you very much for listening, and I invite you to come hear more at poster number 2.
Hi everyone, my name is Anna, and today I'll be presenting my project on sex dimorphism of human astrocytes and their response to viral infection. This work was completed in the Foster Lab. So as you may or may not already know, astrocytes are essentially these star-shaped glial cells that are found in the central nervous system. And essentially, we are focusing on one function, and that is their immune response function. And an important point to note from our background is that astrocytes demonstrate something called sex dimorphism, which is a difference between sexes in both morphology. So as you can see between these two images of male and female astrocyte cultures, and also gene expression. So in the following plots, we have single-cell RNA sequencing data, which shows us that there are 5 subpopulations within astrocytes. And that demonstrates a concept of heterogeneity, which is essentially individual differences between cells. Now, in order to confirm and verify this data, we had to look at this on a proteomic scale, because RNA doesn't necessarily always equal to protein, and also proteomic level can help us understand more about phenotypic changes. And so this is what this heat map shows on the right. As well as this, it introduces the concept of sex dimorphism. As you can see, both the female and male subtypes. Now, onto our methods. Essentially, it was split into 2 parts. So we had a tissue culture and a sample prep. So in a nutshell, basically, we thawed our cryopreserved cells, and then we seeded them and infected them with 2 conditions. So we had an infection condition and a control condition, each having multiple time points of infection. For instance, we had a 16-hour, 24-hour, and so on. After this, we were able to prepare samples by doing a single-cell suspension, lysing them, and then having an incubation overnight with trypsin. And then after our samples were ready, we were able to load them on the liquid chromatograph mass spectrometer. And then after this spat out some data for us, which I'll be discussing below. So first we have this PSA plot. These 2 are on the left. And essentially what this shows us is there is a significant change in the proteome after we infected the astrocytes. And you can see this by looking at the F1 and the M1 controls compared to the infection clusters. So according to their corresponding colors, they have more of a change the longer that the infection period lasted for. Now, on the right, we have feature plots of viral proteins, which basically confirms that the changes in proteome that we see in the PCA is due to infection and not just some other environmental changes. So for instance, the 2 plots on the right, they show that the male samples were able to basically decrease in abundance, while the female samples plateaued. So that tells us that maybe the male samples decreasing means that the astrocytes are essentially fighting back or having some kind of degradation of viral protein mechanism that is not present in the females, which shows us that there's some kind of difference in their response to infection. Now, to further this, we were looking at the functions of the proteins that were within the cell. So within the astrocytes, we have these immune proteins. So for instance, the 2 plots on the top, the IFIT1 and IFIT5, these are immune proteins that essentially they have abundance that increased in both the male and female samples. And this shows us that there is some kind of immune response that's activated, some kind of pathway that's activated. And we can see that in the 48 and 72-hour mark, this is more abundant in males than females, which shows that there's some kind of sex dimorphism. Now, just to conclude, our data has shown that there is significant change in the cellular protein following our viral infection. So our next steps will be to do single-cell proteomics on this infection data, integrate this with other omics such as transcriptomics and lipidomics, and then we can look into some targeted therapeutic strategies for neurological disorders. Thank you very much.
Hi everyone, I'm Arya, and this summer I worked with Mend the Gap team at UBC in the Chung lab to assess the neurite growth at the interface of 2 hydrogels for spinal cord injury repair. Spinal cord injury is a lifelong disability and is also quite prevalent. And basically, in this condition, the connection between the brain and the body is compromised, leading to loss of movement and sensation. Mend the Gap is a project that aims to restore this lost connection by developing an injectable hydrogel that would fill in the lesion site and promote neural regeneration. One main challenge in this approach is being able to extend growth from surviving neurons around the injury into the biomaterial that we injected, which is what I looked into using the microfluidic SCI chip shown on the left, where the wells signify the area surrounding the injury and the channel mimics the injury site itself. I looked into neurite growth specifically at the interface of two hydrogels, one of which was injected in the well of the SCI chip and had a composition of 3% gelatin methacrylate with glutathione, which we've previously seen in our lab to give good growth. And the second gel was added to the channel, which had additional test peptides functionalized to the base gel. I mainly looked into two peptides. One was DNSP11, which is a fragment of glial cell line-derived neurotrophic factor, and the other was ICVAV, which is a cell-adhesive peptide derived from laminin, both of which promote neural regeneration. Rat cortical explants or brain tissue was extracted and seeded in the gel in the well. Then the gel in the channel was added. The entire system was photocross-linked, cultured for 5 days, and then imaged in confocal microscopy. These are the results from microscopy. The bright irregular masses that you see here are the body of the tissue, and the thin lines extending from it are the neurite extensions. The red region marked here is the hydrogel interface region, which we analyzed for neurite growth in each condition. However, you can see that there is a lot of variation in the placement of the explant with respect to this hydrogel interface region, which is what caused inconsistencies in the neurite length required to reach the hydrogel interface. And hence, there was no definitive conclusion that we could draw from this experiment. However, in the future, we plan on using dissociated cortical explants to standardize the number and distribution of cells, and also work on a script to quantify the neurite density. Thank you.
Hello, everyone. Can you hear me okay? Okay, perfect. So hi, everyone. I'm Chris. I'm currently a fourth-year pharmacy student. And this summer, alongside Dr. Heather Leach, we worked on the effect of cardiac medications on clinical outcomes in MDS. So MDS, or myeloid dysplastic syndrome, is a group of clonohematopoietic malignancies leading to cytopenia. The most clinically relevant cytopenia is anemia. Unfortunately, treatment options and survival are quite limited in our MDS population. So most MDS patients will require red blood cell transfusion at some point in their treatment course. This can result in transfusion dependence leading to iron overload, which is a clinical concern once about 20 units have been received. Iron overload can contribute to cardiac morbidity, sitting at the most frequent cause of non-leukemic death. And so iron overload from the repeated transfusions can exceed the transferrin binding capacity, resulting in NTBI, or non-transferrin bound iron, leading to oxidative stress, which is usually reflected with a TSAT over 80%. So NTBI can enter L-type calcium channel blockers, or correction, L-type calcium channels, as well as ZIP14. Both are present on cardiomyocytes, and then the ZIP14 is on hematopoietic stem cells. And so our question this summer was, does the use of calcium channel blocker improve cardiac and other endpoints in lower risk MDS? So we did a retrospective chart review with data pulled from the EMR. We have 149 transfusion dependent patients with lower risk MDS diagnosed between 1985 to 2024. We used the SPSS software to do a lot of our data collection as well as the analysis. And so I know there's quite a bit going on this slide, so to kind of orient everyone, let's start with the bar graph on the top. So MDS patients with TSAT over 80%, again, that's a reflection of the oxidative stress. Those receiving calcium channel blockers had no cardiac events. And if we look below, calcium channel blocker users also had a longer time to the first cardiac event, suggesting NTBI protection. And if we look to your right, time to the first red blood cell transfusion was also longer in calcium channel blocker users. With that said, we can say that calcium channel blockers may protect against iron overload-related cardiac events and delay transfusion dependence by blocking NTBI, leading to NTBI protection. Thank you.
Hi everyone, my name is Claire and I'm the summer student with the Kim Lab. So this summer I looked at the role of filament A in the actin cytoskeleton, particularly in the context of platelet activation. So let's start with the basics. Platelets are small anucleate cell fragments in the blood that play a role in hemostasis. So when a blood vessel is injured, platelets adhere to the damaged site, activate, and aggregate to form a plug that prevents blood loss. So in their resting state, platelets are small and discoid, but upon activation, they extend filopodia and lamellipodia and increase their surface area to form a clot that prevents blood loss. So this actin reorganization involves the polymerization of actin filaments and the reorganization of the cytoskeletal network. Now this brings us to filament A. Filament A is a large dimeric actin-binding protein that cross-links F-actin filaments into orthogonal networks, providing mechanical strength and structural stability. But more importantly, it acts as a scaffold that connects actin to transmembrane receptors and signaling molecules. So in platelets, filament A stabilizes the actin cytoskeleton during shape change, and it connects actin to transmembrane adhesion receptors, which is critical for adhesion under high shear stress, like in arteries. Essentially, without filament A, platelets would become unstable and wouldn't be able to properly adhere or aggregate, which leads to bleeding disorders. So LTC4 is a lipid signaling molecule that is known to play a role in platelet activation. And our hypothesis was that LTC4 is involved in the filament A activation pathway. So to study this, we compared control platelets to filament A knockout platelets. And the control platelets are seen in the left 3 lanes, and the filament A knockout platelets are seen in the right 3 lanes. And what we looked at here was levels of PAC2 phosphorylation, because PAC2 is known to play a role in cytoskeletal reorganization. Now, when we induced these platelets with a thrombin and LTC4, we saw an increase in PAC2 phosphorylation in the control platelets, but this same result was not seen in the filament A knockout platelets, which supports our hypothesis that LTC4 is in fact involved in the filament A activation pathway. Thank you so much for listening to my presentation. And if you'd like to learn more about filament A, or if you have any other questions, please come visit me at my poster, number 6. Thanks so much for sticking around.
Hi everyone, my name is Claire. I'm working in the Gordon Lab, and today I'll be presenting on the role of odorant receptors to eucalyptal-driven avoidance in Drosophila melanogaster. Eucalyptal is known to reduce attraction to mosquitoes to human odors. We hypothesize that this avoidance behavior likely involves the olfactory system consisting of the antennae and maxillary palps that contain sensilla housing olfactory sensory neurons. Fruit flies are a widespread model organism for genetics and neurobiological research. There are 2 main families of olfactory receptors that have different odorant profiles. Those are known as the odorant receptors, ORs, and the ionotropic receptors, IRs. In both vertebrates and invertebrates, odorants bind to ORs in cilia or dendrites of bipolar OSNs. This sends axons to the brain and connects with second-order neurons in glomeruli of the olfactory bulb in humans or the antennal lobe in insects. My research objectives were to determine if eucalyptal avoidance in Drosophila melanogaster is mediated by olfactory input, and to identify the specific olfactory receptor family and tuning receptors required for this behavior. A 2-choice assay was used to quantify the aversion of genetically modified flies. We tested different mutants to differentiate between smell or taste, to compare the roles of IR and OR, and to identify specific tuning ORs. Flies were placed on a plate with eucalyptal on one half and no odor on the other. Behavior was recorded for 10 minutes, and the data was analyzed to quantify the avoidance index. Wild-type flies, also known as white 1118, exhibited a strong avoidance to eucalyptal. Removal of their antenna, the main olfactory organ, resulted in a loss of avoidance, whereas poxin flies that are mutated to be taste blind had little to no effect on avoidance. A mutation in the ORCO gene essential to all OR function showed a significant loss of avoidance. IR8A mutant flies still showed a significant loss, but not to the same extent. The corresponding heat maps for each graph are pictured on the right to visualize the average position of the fruit flies in log frequency. Several OR mutants that are known to respond to eucalyptal were also tested. Our results indicate that OR10A is required for this avoidance behavior. To confirm that this behavior was olfactory-driven rather than touch-dependent, the experiment was replicated with a mesh barrier. This reinforced our results and showed that OR10A is important. In conclusion, eucalyptal avoidance in Drosophila melanogaster is driven by the olfactory system with OR10A required for this behavior. The IR family plays only a minor role, and the behavior as a whole is not dependent on touch sensation. Future directions include testing more additional QORs, testing the sensitivity range of fruit flies to eucalyptal, and confirming OR10A activation using calcium imaging. Thank you very much.
Hello, my name is Danielle, and I'm a student in the Horwitz lab this summer, and the focus of my project has been on identifying commonalities in autoimmune diseases after infection with latent marine gamma herpes virus 68. So just some background into autoimmune diseases. The current etiology for how they develop remains unknown, but one proposed method is this two-hit model of autoimmunity, where early in life a person experiences this immune priming phase, where their immune system takes on this more pro-inflammatory form with less regulatory immune cells, and this can be characterized by things like genetics, sex, or existing disease. And then later on in life is the second hit or disease initiation phase, where these pro-inflammatory cells can migrate to target tissues, and this can be caused by environmental factors like smoking, vitamin D deficiency, or what our lab looks at, which is viruses. The specific virus we focus on is Epstein-Barr virus, which has been correlated to various types of autoimmune diseases. EBV unfortunately only does primarily infect humans, so to study this in mouse models, we use a common homolog, which is gamma herpes virus 68. The two diseases I'm interested in comparing is multiple sclerosis and psoriatic arthritis, which had different types of target tissues, with MS focusing on the CNS organs and PSA focusing on the joints and skin. To study these in mice, each disease has their own induction models, with MS using the popular EAE model and psoriatic arthritis using the Mannin-induced PSA model. Both of these induction methods can be done both passively as well as actively. What our lab has used for many years is this active form of EAE, where mice are infected with virus, left for 5 weeks to allow viral latency to be established, and then they are injected with the myelin oligodendrocyte glycoprotein, or MOG for short, which is a self-antigen, and this is emulsified and complete Freud's adjuvant to upregulate the immune system. This is also supplemented with pertussis toxin to permeabilize the blood-brain barrier. We then take the CNS organs and the spleen, and we immune profile them using flow cytometry. What I have focused on this summer is using a passive model of EAE, where we use the same active induction model, but instead we take the T cells from donor mice, we culture them, and then we inject them into recipient mice, and again take the organs for immune profiling to assess T cell autoreactivity. Like I mentioned, another model we have tried is using this MIP model, where you inject mannin from yeast into mice once a week for 4 weeks straight, and again, take your organs of interest for immune profiling. What we have seen with the clinical results is that with the active EAE, the infected mice tend to have higher disease severity. We've actually seen the inverse result with our passive EAE, but we don't think this has any clinical significance and rather has to do with the cytokines we've supplemented into our culture media, skewing for Th1 cells, which are blocked by the virus. As you can see in the middle and on the far right, this is our psoriasis and arthritis data. Clearly, we don't see any distinct trends between the virus groups at the moment, but this was our first time piloting it, so we're hoping to expand on that. In the future, I am running 2 repeat experiments of this. And then another thing to look at is consider the involvement that the virus might have in the first hit of this 2-hit model of autoimmunity. And obviously, the ultimate goal is to study the role of EBV in these types of diseases. So moving into humanized models would be the plan for the future. Thank you. And if you have any more information or questions for me, I'll be at poster number 8.
Hi, everyone. My name is Garen Barr, and I'm a mechanical engineering student here at UBC. I'm specializing in mechatronics, and in the MA lab this summer, I spent my time working on automating the washing process for nanowell plate manufacturing. So, what are nanowells? They're 96 well glass slides that we subdivide into over 700,000 individual wells. These plates allow researchers to isolate single cells and perform high-throughput imaging data-based experiments for studying complex cell behaviors. In manufacturing, as you can imagine, it's essential to repeatedly clean these plates. Initially, this was done by hand. Unfortunately, it was slow and also inconsistent. This degraded imaging and experimental results. So, to solve this, we built a 2-axis gantry robot with a 3-tank array for washing the plates. This 3-tank system was built on our principle of dilution being our solution. The idea is we progressively clean the plates with a first bath where we initially wash off the main amount of residual material from manufacturing. We rinse it again in a second to give it a good clean, and then we polish it off in a 3rd tank. Each of these tanks has its own 1-micron filtration system, which is powered by an air-driven pump so that we keep our electronics nice and far away from any liquid solutions. And so this system is automated by the 2-axis gantry that you can see on screen here. How it works is you load the plates in, and you press start, and you watch the system run. It will move the plates into the first tank, wash them for a set amount of time, raise them back up, shake them off to remove any excess dripping, and then move to the next tank. It repeats this one more time before it finishes the cycle. This runs completely unattended, which has saved us a ton of time, allowing us to move on to other projects in the lab as we continue manufacturing. And so here are the results. By the end of the summer, we had automated the entire process using this robot that you can see on the left. And on the right is one of the nanowell plates that we've washed. As you can see, it's incredibly clean and provides fantastic imaging qualities for researchers and their single-cell experiments. Thanks for listening, and I'll be happy to show you a video of the robot in action at my poster.
Hey everyone, I'm Javad and I'll be presenting my work in Dr. Tanit's lab regarding the role of telomere-1 in mammalian development and cell ECM adhesion. Proper animal embryonic development requires a coordinated contact between the cell and the outside environment, or the ECM. Integrins are the main ECM binding proteins. One of the most, which can transmit signals from outside the cell to the inside, called outside-in activation, or from inside the cell to outside, called inside-out activation. One of the most important integrin binding and activating proteins is talin, which is of interest in our lab. Recent studies have revealed a novel protein called talin rod domain containing protein, which shares high sequence homology to talin's R7, R8 domain. Despite these sequence homology, however, talin rod 1 is involved in actin bundling. Functional analysis of this protein also shows that it's involved in cell protrusion formation and cell migration. Based on this data, our lab hypothesized that this protein is involved in animal embryonic development and cell ECM adhesion. And to test this, we generated the first knockout mutation of this protein in mice using CRISPR-Cas9. Our genotyping data shows no homozygous adult mice, showing that this mutation is embryonically lethal. Additionally, determining at which embryonic stage this lethality happened is still under progress and requires more data. So at this point, we know that this mutation is involved in embryonic development, but we also want to understand the cellular defects caused by this mutation. Specifically, at sites where integrins bind the actin cytoskeleton of the cell, called focal adhesions, or FAs. And to do this, we derive mouse embryonic fibroblasts, or MEFs, from these embryos and stain them with phalloidin for actin and pixelin for FAs, and analyze this data using a custom-made MATLAB program. Our data shows a small but statistically significant decrease in number of FAs and cell protrusion, as well as an increase in FA area. The overall FA size distribution, however, is similar across genotypes. Now, this is surprising because we know that this protein is involved in embryonic development. Additionally, prior research in our lab involving disruption of talin's function shows a pronounced effect on these factors. So this could mean that tail and rod modulates cell ECM adhesion and animal embryonic development through a distinct mechanism, that of talin, which is very exciting, and we're looking forward to doing more research on this protein. If you want to find out more about my research this summer, you can come talk to me at poster number 10. Thank you very much for listening.
Hello everyone, today I'll be presenting the project I worked on in Dr. Hugh Kim's lab, investigating the role of platelet factor 4 in MEK-ERK activation and interleukin-33 production in asthma. Asthma is a chronic inflammatory lung disease that affects over 3 million Canadians. Cytokines including interleukin-33, IL-33, IL-6, and IL-8 drive inflammation in asthma. In this study, we focused specifically on the role IL-33 plays in this process. Platelets also increase inflammation in asthma by releasing a pro-inflammatory chemokine, platelet factor 4, during platelet activation. In a papain asthma model, PF4 knockout mice exhibited less eosinophil recruitment compared to that in wild-type mice, suggesting that PF4 contributes to eosinophil recruitment. We hypothesized that PF4 promotes asthma by increasing expression and secretion of IL-33 via the p-ERK pathway in epithelial cells. We seeded plates with the A549 cell line, starved the cells on day 1, and treated with PF4 on day 2. After 1 hour of treatment, we prepared for Western blot. The aims of our study were to compare via histology the degree of inflammation and IL-33 staining in the lung tissue of PF4 deficient mice and controls, compare the spatial localization and staining intensity of PF4 and IL-33 in human lung tissue from asthmatic patients and controls, and finally measure the expression of p-ERK protein levels in A549 cells cultured in the presence of recombinant PF4. Representative IF images of human control and asthmatic FFPE lung sections stained for DAPI, PF4, IL-33, and ECAD, and the quantification of IL-33 and PF4 staining in human control and asthmatic lung sections showed significant difference in percent IL-33 present in control and asthmatic lungs. Through scoring representative H&E images of wild-type and PF4 knockout mice lung samples, we found significant difference in the degree of inflammation in treated and untreated conditions. Further, representative IF images of wild-type and PF4 knockout mice frozen lung samples stained for DAPI, PF4, IL-33, and paired with quantification confirmed that experimental asthma upregulated IL-33 expression in wild-type mice. Results from Western blot showed that PF4-treated A549 cells increase the p-ERK protein expression, indicating the pathway has been activated. Our conclusions were experimental asthma upregulated IL-33 expression in wild-type but not PF4 knockout mice. PF4 and IL-33 expression was upregulated in human asthmatic lungs. PF4 stimulation of A549 increased IL-33 expression, and PF4-treated A549 increases p-ERK protein expression. Future studies on the effects of PF4 on lung epithelial cells may further clarify the relationship between PF4 and IL-33 in asthma. Thank you.
Hi everyone, my name is Kayla and I had the pleasure of working in the Cote lab. My project was on optimizing a flow cytometry panel for hormone receptor assessment on immune cell subsets. So we know that hormone receptors are found on peripheral blood mononuclear cells, allowing hormones to modulate immune responses. Using the example of inflammation, when estrogen binds to estrogen receptor alpha, a pro-inflammatory response results, while when estrogen binds to ER beta, an anti-inflammatory response occurs. So given this influence of hormones on immune function, it's important to study hormone receptors in assessments of cellular immune health. However, there's currently no standardized method to study hormone receptors in PBMCs while simultaneously characterizing immune cells. Thus, my work was to optimize a spectral flow cytometry panel for assessing hormone receptor expression in B cells, plasma cells, NK cells, and monocytes. Note that 2 other students in our lab have already optimized this panel for T cell characterization. This panel will be used to assay PBMCs from our cohort of women living with HIV from the BCC3 study to assess associations between hormone dysregulation, inflammation, and cellular aging. Now, we chose spectral flow cytometry for its capability of multiparametric analysis and ability to include more parameters compared to conventional flow, which is necessary to study hormone receptors simultaneously with other characteristics of immune cells. So this is the proposed procedure that I was optimizing. First, we isolate and cryopreserve PBMCs from blood samples. Then we stain our PBMCs with fluorophore-labeled antibodies specific to cell antigens. Our extracellular antigens are the identity markers for B cells, plasma cells, NK cells, and monocytes, and markers of cellular activation, senescence, and exhaustion. Our intracellular antigens are the hormone receptors. Next, we run our samples through the spectral flow cytometer. In our first analysis step, we must separate the different fluorophore colors so we can distinguish between our cell markers. And finally, we can plot our cells based on the expression of these markers. So this is an example of results from one of my studies. Here, I was obtaining single-stained controls, which are cells stained with only one fluorescent antibody. In spectral flow cytometry, we use these to obtain the fluorescent signature for a fluorophore, which is what you can see in panel B for the fluorophore RB705. These signatures are how we can distinguish between fluorophores when we have samples stained with multiple fluorescent antibodies. So on the graphs, the x-axis is essentially wavelength and the y-axis is the intensity of fluorescence. Panel A shows an unstained sample, and as we expect, we see only low levels of autofluorescence. Panel B shows the single stain of anti-CD38 with fluorophore RB705, and we can see a distinct fluorescent signature that looks much different than the unstained and with higher levels of fluorescence. So this is a clean fluorescent signature that can be used to measure the abundance of CD38. If you're interested in learning more about our results or about our panel, you can come visit me at poster 12. Thank you very much.
Hello everyone, my name is Lucas. I'm with the LIM Lab and I'll be telling you about my research on anthracotic pigments. So anthracotic pigments are black carbon deposits which accumulate due to inhalation of pollution, coal, or smoke. These pigments may also be involved in the contribution of lung cancer potential, as studies show that a larger number of cells containing oncogenic mutations are associated with these pigments. And so there's a need to understand the role of anthracosis in the bigger picture of pollution and lung cancer development. So the goal of this project is to develop a method to identify and quantify these anthracotic pigments using a program called QPath. We can then apply these to many tissue slides to characterize anthracosis and its relationship with other clinical and molecular variables, particularly with never smoking patients. In order to quantify anthracosis, we developed a method called SHADE. Here's how it works. So first we have a surgical tissue slide. We'll then detect all the tissue within the tissue slide, which is overlaid in green. Then we find all the dark pixels within the tissue area, which is overlaid in yellow. And then we remove all the dark cellular features that can be stained by H&E, and the result is shown in the red overlay. And then lastly, we remove any other dark tissue features, and the final result is the anthracotic pigments, which again is shown in red. And next, on 134 cases of never smoking lung cancer patients, we use a generalized linear model to characterize the relationship between anthracotic pigment and various clinical variables. And we found that increasing age and the upper lung lobes had a significant relationship with higher amounts of anthracosis. And this finding for higher anthracosis to be present with increasing age also mirrors similar results from other studies of anthracotic pigments. And this also makes sense as people accumulate different exposures in their lungs over the course of their lifetime. Similarly, the association between anthracotic pigment and the upper lung lobes also mirrors findings from other studies. However, the clinical significance and mechanism behind this remains unknown. We also found that cases with higher levels of anthracosis were more likely to be of a micropapillary subtype. Now, this is clinically important because the micropapillary pattern is known to be more aggressive with a worse prognosis, especially in early stage disease. So to summarize, anthracotic pigments might be involved in lung cancer development. And to investigate this, we developed a method called shade to quantify these anthracotic pigments. And then applying shade, we found that age and the upper lung lobes were significantly associated with higher levels of anthracosis, which was similar to other studies. And however, something new that we found was that the micropapillary pattern was significantly associated with higher levels of anthracotic pigment, which again is important because this pattern is especially known to be more aggressive. If you want to learn more, you can come find me at poster 13. Thank you.
Okay. Hi, everyone. My name is Michelle, and today I'll be presenting my work in creating cell surface targeted enzymes for blood group conversion to universal O-type blood. This work was done in the Kazaka Dathu lab in collaboration with the Withers group. Now, as we all know, blood transfusions are essential in healthcare. Though there are over 30 known discrete blood types, the A and B carbohydrate-based antigens of the ABL blood system are the most immunogenic and clinically significant. Therefore, to prevent incompatibilities that may occur in transfusions, ABO blood types between the host and donor must be matched with careful blood typing. In emergency situations when the host blood type is unknown, O-type blood is often used because it is absent of surface antigens and will thus not trigger immune responses. O-type blood is therefore in high demand. Now, recent research has shown that the A and B antigens on RBC surfaces can be enzymatically converted to type O. However, this practice has not yet been adopted clinically. More active and cell surface specific enzymes are required for clinical translation. This is where my project comes in. We hypothesize that the conjugation of blood group cleaving enzymes with cell surface binding antibodies will enhance antigen cleavage efficiency on the cell surface. Now, to test this hypothesis, two steps must be followed. First, we must synthesize the antibody enzyme conjugate. This was done by reacting an anti-A antibody with an A cleaving enzyme. Once produced, the antibody enzyme conjugate was then tested in human blood samples to see if we could increase cleavage efficiency when using the antibody enzyme conjugate in converting A-type blood to O-type blood. On this slide, you'll see graphs showing the median fluorescence intensity measured through flow cytometry of the antibody enzyme complex, looking at whether this complex is able to first bind to the surfaces of red blood cells, shown in the graph on the left, and second, cleave the A antigens on the red blood cell surface, shown on the graph on the left. The blood samples were incubated with PBS as a solvent, as a control, the antibody alone or the antibody enzyme conjugate for 45 minutes at room temperature each before the secondary antibodies were added. On the right, surface binding was measured by attaching a secondary antibody containing a fluorophore to the antibody or antibody enzyme complex. Higher MFI thus indicates that the antibody is able to bind to RBC surfaces. As we can see, comparing the leftmost bar or the control condition to the rightmost bar, the antibody enzyme conjugate condition, we see an increase in MFI from around 100 AU to around 1200. This indicates that the conjugate is exhibiting cell surface binding properties. On the right, and A blood group cleaving properties were measured by using an anti-A antigen labeled with APC. This indicates that upon cleavage, the MFI should be reduced. Once again, comparing the leftmost bar on this graph to the rightmost bar, the control to the antibody enzyme conjugate condition respectively, we see a stark decrease in MFI from around 22,000 AU to around 90. This indicates that the conjugate is able to cleave the A antigen. Now, two conclusions can be made from this data. First, the creation of an antibody enzyme conjugate does not appear to interfere with the ability of the A antibody to bind to the surfaces of RBCs. Secondly, the antibody enzyme conjugate is able to still successfully cleave the A antigens located on RBC surfaces. Further studies to expand to other blood group cleaving enzymes are ongoing. Thank you so much for your attention. If you have any questions, please come find me at my poster session later today.
Hi everyone, my name is Minvi and I'm working in Persdal Lab under Dr. Liwa Hao. My project is modified recombinant clotting factor 10 to improve clot busting. I'm sure many of us know someone, whether it be friends or family, who has suffered from a heart attack or stroke. The current clot busting therapies use recombinant tissue plasminogen activator, which converts plasminogen to plasmin. However, therapeutic doses of TPA are much higher than what is found physiologically, resulting in a loss of localization and increased risk for systemic bleeding. Therefore, our objective is to reduce the dose or eliminate our TPA. As fibrin is cleaved by plasmin, C-terminal lysines are revealed to which TPA and plasminogen can bind. This both accelerates and localizes the clot busting process. Recent studies have revealed that factor 10 from the coagulation cascade can help generate even more plasmin. When bound to anionic phospholipid and cleaved, TPA and plasminogen combined, accelerating fibrinolysis. This is called the auxiliary cofactor model. From this concept comes my project, factor 10W beta. Its beta fragment is removed to reveal an additional C-terminal lysine for binding, and it has a histidine 57 to tryptophan mutation in its active site, which inhibits coagulation and enhances fibrinolysis in plasma. An ELISA was first conducted on 11 factor 10W beta mutants, with 3 displaying factor 10 expression. Then a clotting assay was performed. Factor 10 deficient plasma was incubated with factor 10W beta for 1 minute. Then innovin was added and prothrombin time was measured. Of the 11 selected factor 10W beta mutants, none displayed any clotting activity in plasma. This, in conjunction with our ELISA data, confirms that factor 10W beta has factor 10 expression and limited coagulation function. Just to summarize, current therapies for heart attacks and stroke use rTPA, which can cause systemic bleeding. An alternative, factor 10W beta, was created based on the auxiliary cofactor model. Selected mutants were shown to have factor 10 expression and no clotting activity. Those with higher factor 10 expression will later be used to form stable cell lines for future use in vivo and in vitro studies. As a co-op student, I'm very excited to be working on this project for the next 4 months to show that factor 10W beta has potential to become the next generation thrombolytic agent. Thank you.
Good afternoon. My name is Nastrin Davulu, and I'm an undergraduate researcher in the Hydrid Lab. Today, I'll share how we're working to improve gene editing strategies for cystic fibrosis by combining CRISPR-Cas9 with lipid nanoparticle delivery in bioengineered lung models. Cystic fibrosis, or CF, is a genetic disease in which thick, sticky mucus builds up in the lungs, making breathing challenging. It's the most common fatal genetic disease among Canadian youth. And while current treatments can ease symptoms, there's still no cure. CRISPR-Cas9 gene editing offers the possibility of correcting the underlying CFTR mutation in the CFTR gene, but the biggest challenge is delivery. That's where lipid nanoparticles, or LNPs, come in. These tiny fat-based spheres can carry genetic tools into cells. In CF, our goal is to get them past the mucus barrier and into airway cells where they can repair the faulty gene. To identify the most effective LNP, we tested 5 formulations, A through E, on patient-derived bronchial cells at 2 doses. The green signal comes from EGFP, expressed only when delivery is successful. As you can see, formulation D appeared to produce the brightest signal at both doses, suggesting the highest delivery efficiency. To assess gene editing, we measured changes at the HBRT gene in the same patient-derived cells. Here you can see that LNPD achieved the highest editing efficiency at both doses, matching the strong EGFP delivery we saw previously. To test these therapies in a system that mimics the human lung, we use 3D bronchial models that we build in the lab. These models replicate the 2 key layers of the airway, a ciliated epithelium on top and a fibroblast layer underneath. And because they're made from cells from CF patients, they naturally develop a thick mucus layer over time. This setup lets us test our LNP treatments in realistic conditions, showing whether they can penetrate the barrier and reach the target cells where gene editing needs to happen. To see whether we could improve delivery through the thick mucus barrier, we pre-treated the models with mucolytics, which help break down the mucus. Before adding our lead formulation, LNPD, we tested several options and found that pre-treatment increased editing efficiency, with the best conditions reaching around 18%. For context, restoring even 5 to 10% of normal CFTR mRNA can be enough to bring lung function back to normal, so these results are promising. Going forward, we'll further optimize our lead formulation, LMPD, then apply it to directly edit the CFTR gene. We'll then use Using chamber assays to confirm whether this approach restores CFTR function. Thank you for your attention, and I'll be at poster 16.
Hi everyone, my name is Naomi. This summer, I've had the pleasure of working at the McNaghney Lab, where I've been exploring the role of the early life microbiome on immune development. Although immune development begins in utero, it's really the microbes colonizing after birth that influence its education. At this stage, the neonatal immune system looks quite different from an adult's. Rather than launching strong inflammatory responses to all the new commensals and other microbes it encounters, it's actually biased towards tolerance. While this was once seen as being simply immunodeficient, we now understand that the neonatal immune system is adapted to accommodate for all these new exposures. But disruptions to the early life microbiome, such as through antibiotic use like vancomycin, can throw off this early life immune programming, and this can raise the risk for allergic disease later in life. To model early life dysbiosis, we treat breeding pairs and nursing dams with vancomycin drinking water, and we continue this antibiotic exposure on the pups. We then analyze immune responses in these pups at postnatal day 14 and again at week 5 using ELISA and flow cytometry. Recent studies have shown that serotonin is up in the early life gut, but not in adults, and that microbes uniquely present to this early developmental stage might be the ones driving the serotonin production. So, what would happen if we disrupted this early life microbiome? Well, in our vancomycin-treated pups, we saw lower serotonin concentrations in the colon compared to the controls. So, this led us to wonder whether this drop in early life serotonin has any immune consequences, and if so, do these changes stick around? Our lab has previously shown that early life exposure to vancomycin reduces lifelong Tregs, as shown here at week 7. In our current data, we are seeing this drop as early as postnatal day 14 and at week 5 when we looked again, which parallels the drop in serotonin that we observed in the gut. This might show that this Treg reduction occurs a lot earlier than we once thought. So this led us to ask whether dendritic cells and maybe their interactions with naive CD4 T cells at this time are altered, and if this is driving our change in Tregs that we're seeing. Interestingly, we're seeing shifts in our overall dendritic cell populations, but when we look a bit more closely at the subsets, they look similar in frequency, but they might be functionally different. We're seeing altered co-stimulatory molecule expression, perhaps suggesting a functional immaturity in these cells. While we're still yet to confirm the role of serotonin in this early life gut, we think that treating mice in early life with vancomycin can deplete these serotonin-producing microbes. This lack of serotonin at this time might affect our dendritic cell populations, impairing their ability to promote Treg differentiation. Instead, the balance might be skewed towards more effector T cells, and this might increase the risk of allergic disease later in life. Thank you so much for listening. If you'd like to learn more, please come visit my poster at number 17.
Hello, my name is Nathan. My project in the Whale Lab is about modeling crossover interference. So just some brief background. Crossover is when homologous chromosomes physically overlap and then exchange segments of DNA. So you can see on the right, 2 homologous chromosomes lined up, overlapping, breaking, and then reforming with new combinations of alleles. And this is a fundamental genetic process. It promotes genetic diversity and helps prevent species extinction. I'm specifically interested in crossover interference. This is a phenomenon where having one crossover decreases the probability of additional crossovers on the same chromosome. And the purpose or mechanism behind this isn't well understood. So we can imagine a chromosome with no interference having one crossover and after some time having a second crossover. But maybe with interference, the probability of additional crossover is decreased so far that we never make it to that 2 crossover stage. In other words, our chromosome is stuck at having only one crossover. And we're interested in modeling this because we don't really have a no interference reference point to compare to. We can look at a chromosome after interference and see that there's only one crossover. But with no interference, would there still be one crossover? Would there be two 2 crossovers or 3 crossovers, it's hard for us to say. And since we don't know what mechanism causes interference to happen, it's hard to solve this experimentally. Our approach is to simulate different types or strengths of interference, see how the resulting crossover dataset changes, and see what hints that gives us about how interference occurs in the cell. A brief overview of our modeling method: I'm subdividing the chromosome into bins, assigning a probability of crossover to each bin, and then simulating attempts at crossover at different areas on the chromosome. Whenever there's a successful crossover placement, I then modify the probabilities in remaining bins according to whatever type or strength of interference we're studying. From this, we can plot all sorts of metrics that tell us about how interference is affecting the cell. And from this kind of model, we can start to tease out patterns about how interference actually affects the crossover process. For example, on the right here is a distribution of the distances between pairs of crossovers, with the black line representing no interference and the red line representing the presence of interference. We can see that crossovers become more evenly spaced out based on interference alone. And from these kind of plots, we're hoping to get more hints as to how interference actually occurs, how it affects the crossover process, and how we might be able to see how interference affects actual experimental data. Thanks for listening.
Hi everyone, my name is Puneet and I'm from Conway Lab. And today I will be talking about the molecular pathways regulating insulin signaling. Over 600 million people all around the world are living with diabetes. And the prevalence of this disease is significantly increasing every single year, making it a major global health risk that needs to be addressed. And what is type 2 diabetes specifically? It is characterized by having high blood glucose levels due to insulin resistance. In these patients, the amount of glucose is almost twice as the normal healthy ranges. These patients are struggling with many complications and symptoms, one of them being cardiovascular disease with heart attack, venous thrombosis, and stroke, further emphasizing why we need better therapeutic targets for patients with type 2 diabetes who have insulin resistance. For my project, I am focusing on 2 transmembrane proteins called insulin receptor and CD248. As the name suggests, insulin receptor is activated by insulin and is involved in many pathways, the most important one being metabolism. And CD248, it was discovered that interacts with insulin receptor and blocks its activity. And therefore, for my project, I am hypothesizing that CD248 directly interacts with insulin receptor, causing insulin resistance and blocks insulin signaling. And to test that, we performed binding experiments shown on the left-hand side, where we immobilized CD248 to the surface, added insulin receptor, and detected insulin receptor. The same experiment was done in reverse, where insulin receptor was immobilized, CD248 added, and CD248 was detected. Data suggests that there is direct binding between these two. But to confirm that the data is confirmable, we also performed microscale thermophoresis, which is a solution-based binding experiment. Also, from looking at the right-hand side, we see that there is binding between CD248 and insulin receptor, further confirming our results. And overall, we are hopeful that we have identified a biomolecular complex that can be used as a therapeutic target for patients with type 2 diabetes who have insulin resistance, or to overcome insulin resistance. And we are hoping to explore the interaction, the functional relationship between insulin receptor and CD248 further, and identify the domains that directly interact so we can target them directly. And finally, have a solution for these patients. Thank you. My poster number is 19.
Hello, everyone. If you've seen any medical drawings before, you must have heard of the term autoimmunity before. But what does it really mean? Autoimmunity refers to a group of chronic conditions in which the body's immune system is mistakenly activated, and thus it attacks itself. Some well-known examples include type 1 diabetes and rheumatoid arthritis. Now, you may be asking yourself, why would our body ever do this? Well, you see, we have enzymes inside of our bodies which break down big proteins into smaller pieces. If those pieces were originally from bacteria or viruses, they cause an immune response. However, our own proteins do not do this. In the case of an autoimmune patient, this system fails, and thus our body cannot recognize our proteins, and thus you get an immune response. Now, one reason why this autoimmunity may happen in the first place is due to a process that my lab discovered, which is known as transpeptidation. We have found that it is possible for a group of enzymes called cathepsins to fuse a foreign and a self-protein together, forming a neoantigen or a Frankenstein peptide, if you will. This may then cause an autoimmune response. So we took on the challenge to inhibit this process without actually affecting the normal activity of the enzyme. And my aim was to find selective transportation inhibitors and evaluate their efficiency. My methodology consisted of digestion assays. These assays included a substrate, which would then get fused together to a biotinylated donor peptide by the cathepsin. This fusion product would then get incubated with avidin FITC. You see avidin binds to the biotin, and then the FITC gives out this fluorescent signal, which we can see on the blots as these black bands. During my research, I found that aspartame is one good inhibitor. As you can see on the image on the top left, as you go from left to right and as the concentration of aspartame increases, the intensity of the bands decrease. Since the intensity of the bands correspond to the number of fusion products, that means that transportation is being inhibited properly. The image on the right just shows that at 10 and 20 millimolar, you have a 60 and 80% inhibition respectively. The image on the bottom left shows that even at high concentrations of aspartame, the digestion pattern of the enzyme is not changing, so the enzyme is still working properly. So in conclusion, my research showed that aspartame is able to outcompete transportation at a high dose. However, it still may be inhibiting normal catalytic activity. Therefore, future work should aim to find more effective inhibitors that work at a higher efficiency but at a lower dose, because currently aspartame is at the millimolar dose, which is very dangerous. Overall, we should also try to aim to minimize the damage to normal activity while maximizing damage to transportation. Thank you for listening.
Hi, my name's Rachel, and I'm from the Hedrich Lab, and my goal this summer was really to develop protocols for liver organ on chips for studying liver toxicity caused by lipid nanoparticles. So lipid nanoparticles, LNPs, are a really growing drug delivery system. Anyone who's gotten a COVID vaccine, you've had a drug delivered to you by a lipid nanoparticle. And previously, many studies have been done with them, but we don't really have great models to study their toxicity. Currently, it's being done in cell lines like mortalized cells and rodent models, and we don't really look like mice. So we need better models, and that's where this comes in. This is an organ on a chip. It basically allows you to mimic a human organ a lot smaller, and you don't have to use mice. So it has places where you put in your organ tissue, and it uses microfluidics and pumps to mimic the lub-dub of the heartbeat and the stress that that would put on an organ. So our goal is to find a way to use this to study liver toxicity from LNPs, even though it's already been done with existing knowingly toxic drugs with great success. So I can tell you if a drug's toxic, but it also matters if it works. And primarily, lipid nanoparticles are being used for genetic diseases in our lab by doing a knockdown or a knock-in or some kind of other genetic editing. So you can see in this figure, as we increase the dose, you kind of have better editing. But a high dose doesn't necessarily mean it's safe. And as you can see, as we increase the dose of the LNP, we do have more cell death, just visually, which is good to know. But what about the actual data? So these are 3 toxicity assays. AST and ALT are enzymes released by the liver when it's stressed, when the cells are stressed. And as you can see, as we increase the dose, you see more cell stress. And same with lactate dehydrogenase, which is in the cytosol, an enzyme, and it's released when the cell dies. So higher dose, yeah, you might have better editing, but we can also see you also have higher toxicity and higher cell death, so you might not want that dose of the drug. So I did all this work in immortalized cells, which are a current preclinical model. And well, the higher doses you saw in those figures, you would not be putting that into primary cells. You wouldn't be putting that equivalent dose into mice or humans because it would completely kill them. So that's a great example of why immortalized cells aren't great preclinical models and why we need better ones like this. So the future steps involve primary cells in both a monolayer and a spheroid, which are also preclinical models. And then we're going to move on to the organ on a chip, so building a little liver in something like this. So we can study toxicity specifically from lipid nanoparticles.
really cool demo. Hi everyone, my name is Sogan. I'm here from the McNagny Lab to present our ongoing research on identifying blood-based biomarkers for better diagnosis of IPF. Now, what is IPF? Well, IPF stands for idiopathic pulmonary fibrosis, and it is an ILD, or interstitial lung disease. And that just means that patients have a hard time breathing in because their lungs become very stiff. Unfortunately, IPF can be very lethal, and it is also chronic and progressive, and it is lethal particularly because it is often misdiagnosed because it looks like a lot of other ILDs. So our goal here is to try and discover more unique blood-based biomarkers to better distinguish IPF from other ILDs. Now, the current literature out there really focuses on molecular biomarkers of IPF, such as surfactant proteins, matrix metalloproteinases, tissue-inhibiting metalloproteinases. However, these are not just unique to IPF, and they actually exist in other ILDs as well, which is why we have the goal of trying to find more unique biomarkers that can distinguish IPF from other ILDs. So our research objectives here are, one, to characterize more unique molecular biomarkers for IPF using cytokine assays, and then we want to pair that with finding cellular biomarkers for IPF using spectral flow cytometry, which hasn't really been done before, and we're hoping that together those two can help us create a more distinct and unique immune profile for IPF to better distinguish it between other ILDs. This is just the same research objectives in figure and methodology form. I won't go into the details, but would just like to note we are very grateful to have the support of Dr. Donson and Dr. Chris Ryerson from the Center for Heart-Lung Innovation to have a very well-phenotyped cohort of patients for this project. Just to have a little sneak peek at the results, more at the poster, we have some preliminary cytokine assay results. This was objective number 1. And we can see that, can you see my cursor? Yes. From the principal component analysis results, we see quite a clear distinction between the ILD patients and then the other controls, so healthy patients, as well as COPD. COPD is an obstructive lung disease, which is the opposite of interstitial, basically. But then when we dive into the ILDs and look at IPF, hypersensitivity pneumonitis, and some other autoimmune ILDs, we don't really see a clear distinction, but IPF seems to be kind of clustering with other ILDs, which we would like to look a bit more into, and that is kind of alluding to the next steps of our project, which is, again, as I alluded to continue analyzing some of the cytokine assay results to compare IPF versus other ILDs in those subtypes I mentioned in the previous slide. And then we, of course, want to continue analyzing some spectral flow data, which I haven't shown here, but it is at the poster, to identify potential cellular biomarkers. And if you're interested in seeing more results, please come to poster number 22. Thank you so much.
Hi, my name is Zerbab, and I'm from the Jeffries Lab. Today I'll be discussing my research on investigating vascular dysfunction in cerebral organoid models of Alzheimer's disease. Alzheimer's disease, or AD, is a progressive neurodegenerative disorder characterized by memory loss, cognitive decline, and eventual death. It affects approximately 10% of people over the age of 65, yet many AD drug therapies remain ineffective. Pathologically, it's marked by extracellular amyloid beta plaques and intracellular neurofibrillary tangles. Because of the presence of amyloid beta and tau, the dominant amyloid beta aggregation hypothesis suggests that accumulation of amyloid beta in the brain causes plaque formation, ultimately resulting in the classical symptoms of AD. However, the theory is weakened by the presence of amyloid beta in asymptomatic individuals and unsuccessful treatment outcomes in clinical trials. Alternatively, the neurovascular hypothesis of Alzheimer's disease suggests that neurodegeneration and amyloid beta accumulation may be due to a dysfunctional blood-brain barrier and impaired cerebral blood flow. While the mechanisms underlying AD pathophysiology remain unclear, gene variants of early onset Alzheimer's disease, specifically the Swedish mutation in the amyloid precursor protein gene, are interesting areas to explore. My work involved creating stem cell clones containing the Swedish mutation and growing vascularized human cerebral organoids, or mini-brains. The knock-in mutation was introduced to cells using co-electroporation with a CRISPR-Cas9 plasmid and single-stranded DNA. Once at least 10% of the cells were GFP positive, they were FACS sorted and cultured separate from the wild-type cells. In addition to that, the vascularized human cerebral organoids were generated by differentiating human embryonic stem cells into vascular and cerebral organoids and subsequently co-culturing them. In figure 1a, you can see a
relatively successful electroporation attempt, and 1B is a gel electrophoresis analysis of the PCR Swedish DNA. The bands at 500 base pairs are the APP gene. Here you can see the differentiation and development of the blood vessel organoids and that of the cerebral organoids. Once the cerebral organoids were at day 29, they were co-cultured with the day 6 blood vessel organoids, and figure 2C shows the day 59 vascularized human cerebral organoid. As you can see, it's about 1 centimeter in diameter now. Overall, my work shows the successful development of prototype vascularized human cerebral organoids grown past day 60 now. The next steps involve using the electroporated cells to establish a mutant stem cell line that can then be used to create the CO models of Alzheimer's disease. Thank you, and I'll be happy to answer any questions at poster 23.
Hi, I'm Charlotte. This is Jess and Navindi, and we're second-year medical students who had the privilege of working on this project during our first year of medical school here at UBC. So we worked on education and screening for sickle cell disease in rural Nepal. So sickle cell disease is an inherited genetic blood disorder which is passed on in an autosomal recessive pattern. And this point mutation actually occurs in the hemoglobin beta chain, resulting in a glutamic acid to valine substitution. Hemoglobin S results in blood cells to change morphologies to become sickled in shape, and this changes the ability to carry oxygen as well as occlude blood vessels and cause organ damage. So common symptoms and complications of sickle cell disease include fatigue and chronic anemia, as well as delayed growth and puberty. They also experience frequent infections, as well as vaso-occlusive pain crises, which causes organ damage. The most common forms of screening and testing is a sickle cell solubility test, and this diagnosis is confirmed by HPLC. The most common treatment options are hydroxyurea, which increases the amount of fetal hemoglobin in circulation, resulting in decreased sickle cell in circulation, as well as supportive therapies for pain control, infection prevention, and blood transfusions.
So we work specifically in the Dong district of Nepal, which is home to the indigenous Tharo people. Although malaria is largely eradicated now, it used to be endemic to the region and has resulted in a higher prevalence of sickle cell in this population. This is because the sickle cell mutation is kind of protective against malaria. Importantly, we also work with our local NGO partner, Creating Possibilities Nepal, which is based out of Kathmandu. So the UBC Sickle Cell Disease Program has been operating since 2015, and we're involved in 3 main initiatives. The first are SCD screening and diagnosis clinics, where we provide these annual camps free of charge to all participants. Next, we're involved in SCD community education. So all the participants at these workshops also attend little educational sessions to teach them about the cause, diagnosis, treatment, and management of sickle cell. This is important because awareness is pretty low in the region, and there's also a high degree of stigma. So this is something we're trying to address. Finally, we've helped develop pain management workshops for community health workers. Here, they go through the module and also our supplemental educational materials to help improve pharmaceutical literacy.
In this figure over here, we have our sickle cell screening results from the sickle cell screening camps from 2016 to 2024. So within these years, we've actually screened over 6,000 individuals, of which there's been about 8.96 average positive screen rate over the years. And what's interesting is some of these individuals do have the resources to pursue diagnostic testing. And from those diagnostic results, we found that about 5% have had positive for sickle cell disease, and then 55% have a positive test result for sickle cell trait, and then about 10% had a false positive screening test. And here we also have our preliminary results from this year, 2025, where we had 187 patients screened at our annual screening camp with 26 positive screening results, which is 13.9 positive screening rate for this year, which is actually higher than our typical rate every year. So in terms of next steps, we hope to use this data to essentially get funding to be able to pursue avenues such as newborn screening, advocacy, pharmaceutical literacy, and pain management education, as this is a disease that's quite prevalent in this population, in the Tharu population of rural Dong in Nepal. And if you have any questions about our presentation, you can see us at poster presentation 24. Thank you for your time.
Okay, so I think it's fair to say that our students did a really fantastic job. So I think this deserves a round of applause. So. Thank you very much for all of your efforts this summer, and thank you very much for enduring the extreme stress of presenting in front of a group. So we really do appreciate that. And please remember a reminder to everyone to please vote for your favorite oral presenter, and the voting does close at 3:30. Now, I think anyone would agree that, and certainly our students would agree, that any success that we achieve rarely occurs without the help of mentors. And Neil Mackenzie, the late Neil Mackenzie, is an individual who was here at the CBR as a postdoctoral fellow and who really did exemplify all of the characteristics of a positive role model. And for that reason, we have an annual Mentorship Excellence Award in Neil's honor. So I'm very happy to ask Dieter Brahma, who was Neil's postdoc supervisor, to come say a few words about Neil and to present this year's award winner. Thanks, Dieter.
Yeah, first, my congratulations for everybody for the excellent talks. And as you, Kim, mentioned, I think without good mentorship, all this good work would be impossible. So most of the time, more we senior guys have to write grants, have to give lectures, maybe we're traveling. So we need really the help of younger people for the mentorship. And most of the time, these are postdocs or senior graduate students. And now I have to say, time's flying. Neil, I recruited from Edinburgh in Scotland when I was giving a talk, and he was really interested to join Vancouver, UBC. And we wrote a grant together, and we got a grant, a CHR, with a graduate student. That was amazing. And that was in 2013. So he came to Vancouver, started his PhD, his postdoc project. This was pretty much focused on proteases and cardiovascular diseases. He was very successful, got papers out, and was really a great guy to have in the lab. Everybody liked him. He had a lot of parties, and everything was great. And second to the science, he was an outstanding enthusiast of the outdoors. He was always, every weekend, he was away. And we made jokes because always every second weekend he came with a blue arm or with a hit on the head or something. So he was a biker, mountain biker, hiker, and everything. And so he really loved it. This was definitely his second love of his life, to be outside in the mountains. So, but then, unfortunately, a tragic stroke. It was New Year 15. He wrote actually an article, sent it to me still just before New Year's Eve, and asked my advice. This was a kind of more philosophical teaching-related article. So I wrote him back, expected some feedback, and he didn't answer. So, and then we looked around, and then I heard from somebody there was an accident up in what's the lake? Joffrey Lake. It's above Pemberton, behind Whistler. So I know that he was there, but of course there's a lot of hikers there, and we didn't know what's happened. But then the news came in. They found 3 dead people, 2 of his bodies, and they fall down supposedly 800 meters of a ice fall in the mountains. And this was really a tragic event. So we lost him. And in of his honor, we created essentially this award, and we hope that this award will last for a long time. and that always younger people get the award, and we are very grateful to you that you are helping actually the younger ones even more, which are presenting today, to give good advice, hands-on advice in the lab. So, I think this year's name, you want to announce it? Maybe don't know for sure where it is. Here, right here, yeah. So my understanding is the awardee is actually not available today. So how do I activate this guy? How do you? Oh, this one, okay. So this year's awardee is Karen Matthews. She's from Mars Lab, also on the 4th floor. But my understanding is she is not here, right?
Video.
Huh? Oh, she's recorded. So she is live here, or is this recorded?
Thank you to the Center for Blood Research organizers and community for this incredible honor. I am deeply grateful to receive the 2025 Neil Mackenzie Mentorship Excellence Award, and I also want to congratulate the other nominees. It's inspiring to be recognized alongside such dedicated and generous people. This award really means a lot to me because mentorship, at least the way I've experienced and tried to share it, has never been about titles or hierarchies. It's been about friendship. It's about building a community where we can grow together, celebrate each other, and be honest about the messiness of science and life. I've been lucky enough to work with people who are not just brilliant scientists, but also generous, funny, and kind. People who have taught me just as much, if not more, than I've ever offered in return. If I've helped someone feel more confident, more capable, or more at home in this space, then that is the true reward. Mentorship to me is really about showing up, not just with advice, but with empathy, encouragement, and even snacks, sometimes wine, when needed. It's listening, learning, lifting each other up, and reminding each other that none of us have to figure it out on our own. So thank you to the friends who nominated me, to the CBR for this recognition, and to all of you for being part of the community that's made this possible. I'm truly grateful.
So congratulations to Karen. And I think we come now to the next part of the day.
And on that note, I think it's really important that we recognize all the nominees. They're in the program booklet, but I think it's really important to emphasize that all of the success of our students and really all of us can be attributed to the wonderful mentorship and guidance that we're fortunate to receive. So thank you very much to all the mentors who not only tolerated our students, but also taught them and nurtured them. So thank you very, very much. Now it's my great pleasure to welcome Dr. Mohit Bhandari to the CBR to give this year's Research Day keynote address. So Dr. Bhandari did not let the threat of labor strife at Air Canada deter him from visiting us from Hamilton, Ontario. Dr. Bhandari is a distinguished university professor and the chair of the Department of Surgery. He is a Tier 1 Canada Research Chair and is internationally recognized as an expert in epidemiology and surgical clinical trials. His awards and accolades are too long, the list is too long for me to mention here, but it is worthwhile noting that he is both a member of the Order of Ontario as well as the Order of Canada. We are very, very fortunate to have him here today. So Dr. Bhandari, thank you very much.
Great, thanks so much. You can hear me okay? Yeah? Back there? Okay. It's been really interesting. I'm a clinician and I'd say clinical researcher, but hearing some of the fundamental research that you're presenting, first of all, the poise at which you're presenting it and the clarity with which you're presenting it is really, really impressive. So a huge congratulations to all of you who've presented, those of you who've participated, and absolutely all the mentors who have been helping, along this process. So I must say, when I got the email, quite frankly, I was a little confused. I said, I'm an orthopedic surgeon from the Department of Surgery. What possibly could I share with this group? And I think there is a message that I felt, and I wish I had heard long time ago. I found this message out pretty late, so I'm going to share with you a little bit about. Just a second here. Okay, yeah, great. So I'm going to share with you a little bit about this idea of what I'm calling, quantum creativity. A bit of a pithy title, but the truth of the matter is it's basically the science of the small things you can do, the small things I've actually done. And so I would put forth to you that everything I'm talking to you about, I've lived it. So this is not a talk of me reading books and science papers and things. This is actually a talk of 56 years or so of thinking through science and thinking about research and moving forward. Can I just ask, I think these slides have the backgrounds all changed. It might be worth it because I see all the slides in white and the whole background's in black. So I don't know what's going on here. They're all white. Sorry, I don't, because the slides will be completely a mess if I don't. These are all, every slide here is, look, the background's black. Every slide, yeah. I think it's probably on the format of the one you put them in. Do you have the original one on your thing? Could you just start that one up, just separate?
Just close this one.
Yeah, just to make it easier. See, the 30-year-old me would have kept going. Not the 50-year-old. I'm going to stop it and actually do it the way I want to do it. Great. Thanks so much. That's great. Okay. So, this is basically what I'm hoping to do. And I'll share with you, this disclaimer, this is not AI. I don't use AI, although I probably should, but I don't. And what I want to get to you with really is this. So you have a new director, right, in the last 6 months? Is that right? And I actually went and I looked at the vision statement. And the vision statement here, and the statement says we want a world-class hub of scientific creativity. Exactly. McMaster University, the University of British Columbia. The words innovation and creativity are everywhere. And the fundamental truth is when everyone is talking about being creative, what does that usually mean? No one's creative. When everyone's talking about evidence-based medicine at Mac, there's a lot of people who just use the terms. And I actually think that we can live it. And I think in our own institution, we've been really focusing on this idea of innovation. But I think, just as you have, and I think appropriately so, Hugh, I think creativity is such an important part, and I really genuinely appreciate that you put it in as one of your key statements, because I think that reminds us all that we should be moving in this direction. We just published a paper at McMaster with a PhD student of mine who is focusing really on a PhD on creativity in surgery. And it's really important, I also think, to define what it means. This is what we think, and we think creativity means that it's the generation of novel and useful products, ideas, and solutions to unsolved surgical problems. But here's the interesting point. Creativity is beyond being novel, as I'm learning. It has to be useful. And so for me, the fundamental shift in thinking, and with most universities, and I suspect you're probably feeling it, is there's a real push for innovation through entrepreneurship. And all this work here, everything is about, okay, what are we doing ultimately to understand processes, but to invent the future? And invent the future is something that's fundamentally useful. And I think that really is what I've been trying to understand for the last several years. Particular definition that I've liked, and I think it probably resonates with most of you, is that creativity is inventing, it's experimenting, it's growing, it's taking risks, it's breaking rules, it's making mistakes, and ultimately when you're creative, you're having some fun. And that, I think, is really interesting. I was in Japan recently, and there was trying to get into this one particular establishment which has 6 people in it, and you cannot get in. It's like you physically can't not log in and to get in, so you have to show up. And we got in after waiting for some time, and we were not disappointed because there was an element, and they call this individual, one of the most creative bartenders in Japan, in Tokyo. And what makes him interesting is that he has all of this, the smoke and the lights and all this stuff. But at the end of the day, the interesting novelty is one thing, but there has to be use to it. There has to be still quality behind that. So behind the smoke and behind the mirrors, there still has to be quality. So good ideas have to be interesting and innovative, but they also have to be useful and fundamental and high quality. This individual is the individual that I think for me was one of the critical, pivotal individuals in my life. And he and I still connect, and we're going to be going for a mountain bike ride, recently, and we're going to be in, Ontario doesn't have mountains, so we just call them trails, but anyways, we're going to go for trail riding. But 25 years ago, he was really looking to move the idea of evidence-based medicine forward into surgery. So he and I met, and Gordon Guide, if you don't know who he is, he's a professor at McMaster University, many accolades, many citations. If you understand what the word H-index means, he has an H-index of about 305. So if your jaw dropped, that's appropriate. It's pretty high. But he was the fundamental founder of evidence-based medicine at McMaster and has become, quite internationally recognized for it. He said back then, when I went to him as a master's student, my second year, he says, would you like, could you be my supervisor? And I was nervous and, really fumbling all over the place. And he goes, well, what do you want to do? I go, well, I just like to defend my thesis and like to get this done. And he goes, okay, what's your goal in life? And I'm thinking, I just want to defend my thesis. I really want to get this done. And I hope you can be it because I can't find a supervisor. That was really the reality of it. And he says, I need you to think big. And, I didn't understand that at that time, but I took it as be productive. And I suspect most of us think of it as, just do lots of stuff. And that's what I did. I published, I don't know, maybe now 1,200 papers, maybe, something like that. And if I look back, and I look back at all those publications, I'm very proud of a lot of them, but a lot of them were just on a treadmill, just going forward. And if I go back to, let's say, the 15, 17 papers that I think would have been impactful, if I just had my resume that said, I've got Lancet New England papers, and that's my resume, I'd be probably very proud. In fact, I would be proud. I am very proud. But then I wonder about the other 1,050 or so of them. What did that accomplish? What did that do? And I actually went back and looked at the amount of time I spent in the process of those papers. It's about 10 years of my life, like 10 actual years of time for those other papers. And you go back and you say, well, what could I have done with that time if I'd focused on not confusing being productive with being impactful? And now it's easy to say. You can say, well, you can say that because you have 1,000 papers. When you don't have one, you have to keep trying to get papers. And I know that game, and I know that the hypocrisy of some of the things that I'm saying. But I'll tell you, no one ever said that to me. They kept saying, just keep writing, keep going, keep going. And in fact, the challenge we have, and when you look at actual literature, and I've been thinking carefully about, really what is this link between productivity and impact? Well, in fact, the productivity of a scientist, the number of things you do and write, and the average impact per paper of the scientist are almost uncorrelated. Almost uncorrelated. So that gets you to back and think, well, maybe we should be really reinvesting in this idea of the idea, and then really thinking about what makes something really, really important. Then you say, okay, well, how do we do that? Well, first is do less. The advice to my younger self is I wish I had done a lot less. I wish I had slowed down a bit, just not be in a rush to figure out what the next year is going to be and just focus on the moment. Enjoy what you've just gone through. Like, this looks like it's been an amazing summer for many of you. And that's what's exciting. When I sit there, I go, wow, what a great place to be. And I want to go back to Mac and say, well, we should be doing something like this. We should be definitely celebrating what you're doing. So I've learned a lot, actually, watching this. And focus on quality over quantity. I wish I'd done that. I really do. Not to say I haven't tried, and I've done things I think are important, and I feel proud of that. But I also think that I rushed a lot of stuff. I just cranked stuff out just because I felt that was what was going to be needed. The future belongs to those who learn the skills. You've got to learn something, but you have to use it in creative ways. So just being creative for the sake of being creative is nice, but you have to have domain expertise in something, I've learned. So you have to know something about something and then think about, how do I now move the needle? Go from, take a big leap rather than incremental leap. How do you do that? Well, we've seen it, right? We've seen it. In Canada, Geoffrey Hinton in Toronto ultimately received the Nobel Prize in Physics for what we now consider basically large language models and learning models and machine learning and artificial neural networks. Anyone know how long ago the paper he wrote that became the fundamental paper for which this was awarded was? How long ago he came up with that idea? Take a guess when Yale went out? 40 years. 40 years ago. So what prevented it from happening? We just didn't have the computing power. We didn't have it. So when you think about, so for him, for many years, it was a lot about moving the step forward until something will happen. But having that absolute stroke of genius, you can call it, to think 40 years in advance, right, is pretty impressive. And I'm not saying that, I'm not saying any of us here are capable of waiting 40 years to see the result. But I think you have to be thinking that way. You have to start training your mind to think that way. And we are in a world of AI, and Jensen Huang, who is the CEO of NVIDIA, who was, basically made a gaming chip, became, what's it, $1 trillion company in the age of AI, because all the companies do now, all the CEOs of all these large language model groups do you know what their number one thing they do? Is they hunt for data and power in space. That's all. They're just hunting for data processing. That's all they do. They don't look for money, they look for data processing. They all go on these programs and say, we're looking for more and more processing power. And he is, in the possession of lots of processing power. But people using AI are ultimately going to be the fundamental game changers. So how are we going to use this appropriately and how are we going to move forward? Now, there are others who say, some of the elite producers, Rick Rubin is definitely one of them, will say, you know what, the problem is AI is reasonable. It looks at everything that's been done, but great breakthroughs have come from people who are unreasonable. So also think of it that way, right? Like, how do we get to the idea? And sometimes because nobody gets it, doesn't mean we should just bury it. We should think, okay, maybe we're onto something. We should really follow it and really try to explore it. But this is the hardest part of research. And here's the challenge I think we all face, all of us face, is asking a question big enough worth answering in your career, in your life, but actually small enough you can take chunks of answering it. And then working your way through it. I saw lots and lots of great stuff today about, you're working through the process. And you can see very systematically how you and your mentor, and clearly the labs here are thinking, and the PIs here are thinking very thoughtfully about what you can be doing. But it's fascinating to think about that. But here is the dilemma we are in, all of us. 45 million publications, 3.9 million patents have been evaluated. And what does it tell us? Progress in several fields is slowing. We're actually seeing impact going downward. Productivity is fine, but the impact of the work we're doing is decreasing, which means more than ever, I think, we have to be reinvesting in something that we haven't been. So writing more and doing more isn't getting us more. So what can we be doing? Right? We have to be thinking about the fundamental C word here is curiosity. How do we get more curious? And then from curiosity, how do we push that forward to ultimately get into creativity? And while intelligence, for sure, in humanity has been going up, for sure, creative thinking is declining. In fact, that decline is steady and persistent. Creativity scores from validated tests of giving divergent thinking creativity, so just how we, the way in which we answer questions, the way in which we come up with novel insights. If you give tests to 5-year-olds and adults, same tests, adults fail because we've gotten so narrowly focused on the answer rather than anything that could be blue sky, almost crazy, because we think, well, that's not going to be feasible. It's not going to be possible. So I'm going to focus on what we can actually do. But when you look at tests of association, and these are these validated tests, when you ask about a problem, and as we go further along in our training, we have actually fewer numbers of solutions to them because we think we have the answer right off the bat. We're basically told, our university system, certainly our medical school system, is there is an answer. And so we don't really care about the variety of answers. We don't really care about how unique your answers are. And we don't worry about whether you know something, that you've pulled something from economic theory and brought it here and you're going to apply it to this patient. That's not the reality. It's what is the answer. And that's a big challenge. In fact, at our own program, so I'll speak to McMaster, we evaluated 80 of our surgical trainees, anywhere from 0, meaning they're a medical student entering first year, so no surgical training, all the way up to being a senior, just about to graduate. And if you look at higher scores, what do you see there? There is a pretty well almost a fundamental, almost linear trend that goes from decreasing in terms of overall creativity scores on divergent thinking. Now, one might argue, I don't want my surgeon to be creative. I don't want my pilots to be creative, and I don't want the military leaders to be creative. You don't want that. So maybe we're doing exactly what we should be doing. But we also have to think, how are we going to move forward in our field if we don't actually ultimately identify ways to be creative outside of the operating room? And in fact, one of the key findings, this paper was published and it was picked up by Harvard Business School because they're seeing it as the other people see it. And our own field doesn't see it, but other people see it as well. This is important. We should be doing something about this. That our residents' abilities to generate original ideas was significantly below average. And you can argue, again, it's our training. Right? These are highly capable individuals. Our training is in many ways supporting that notion. But arguably, you'd say the best first step, the critical first step in anything, if you're moving forward in research, is the idea. So let me ask you this. Let's take a second here. How do you get your ideas? Lots of great ideas here today. What do you do? When do you get a good idea? When does it come? Does it come when you're in the lab working away, or does it come when you're away, or does it come when you're eating, does it come when you're running? When does it come? Anytime, okay. Okay, now we have to narrow it down a little bit. Just a little bit. Yeah. In the washroom. I don't want to ask. I don't want to ask what you're up to and how long you're in, but yes, absolutely, yeah. In fact, a showerhead company funded some of the largest research on keeping people in the shower. So they have all these design showers because they want people, and then they say, you know, majority of your ideas, 3 or 4 people have an idea, a major idea in the shower, which keeps them there. No joking, it was funded. Yeah, it was funded. Anything else? I'll take a couple more. Yes? Would you watch a movie? Sure. Roadblocks. Okay, so tell me about that. What do you mean by that? I love it, love it. I saw someone online raise their hand. I don't know if we can hear that person.
Yeah, back here, maybe.
Oh, sorry. Oh, right there. Sorry.
Yeah, for me, it's usually about 2 or 3 in the morning when my baby's woken me up and I can't get back to sleep.
There you go. Okay. So, but the point is, thank you. That's great. So the point is, they come from lots of spaces, and the truth is, all the skills in the world won't matter if you can't come up with good ideas. So you can have the most beautiful, procedures in place. You can execute them beautifully, but if it's the wrong execution, that's a problem, right? Poor ideas perfectly executed will always result in a poor outcome. So we've got to get to something that moves us forward. And what holds us back is, I think we're seeing it. We're actually getting data at our own institution that, we're not learning to be original. So what does that actually fundamentally mean? So I've suggest anyways, that there's a lot more than 3 steps, but I'm suggesting that there are a few steps. I've taken each one of these. I do this, and I'll share with you what I do, and it seems to have worked for me, and it's helping me. Is 3 simple steps I think you can do. And I think of it this way, to ignite it. So the first thing is you have to do, you need time. You need to get a high quality network around you, and I think we can speak to that. And then you have to find useful use of that time, and how you think around how you're going to use that time you have is fundamentally important. You got to create time. You have to create time. How many of you actually take truly protected time in your day for something that's outside of your work? It's slated in, it's non-negotiable. How many of you take any time at all? Okay, that's awesome. May I ask how long? Okay, great. So 2 hours is really, that's great. So that's a day a week roughly that you're doing that. How has that worked out? Would you ever give it up having started? How long ago did you start that?
For me, it's just climbing and I don't know, it's exercise. It's kind of small problem solving. It's not every day. It's maybe every other, every 3rd day. I did put in all my presentations a thank you to the climbing gym, so I'd say it's pretty important.
Superb. If I could just put an anecdote, Adam Ondra, you know who Adam Ondra is, others might not, but he's like the climber's climber, would that be fair to say? He's pretty respected. And he had a quote, we had a quote where he said, why do you climb? He goes, I climb for the sake of climbing. And because he climbs for the absolute joy of doing it, it's made him the best in the world. That'd be pretty fair?
I'd say certainly up there.
Yeah. Okay, fair enough. We're going to argue on him, but he's pretty good, right? Pretty good?
Yeah.
Okay. Good. Whew. Okay. But there's a fundamental importance to scheduling nothing. And you'll see this, and again, this is the cliche storyline. Look at all the fancy people; they're all, geniuses, or they're all millionaires and billionaires, and therefore there's a positive, bias. So I get that. And there's a lot of people who think nothing happens for them. I get that too. But I do think there's a fundamental truth to thinking about spending time in a moment, and I'm using the word thinking, but I think you're right. Whatever you feel separates you and gets you in the moment of something, I think is really fundamentally important. Dali and Edison used this approach, which they called it like it's just post-sleep. So I don't know if any of you have had this happen where you're about to nod off. I had it a lot this morning on the plane. You're just, your head bobbing and you're just trying to keep up. And then, you have this moment where just before you go to sleep, it's called N1. The first stage of sleep is what really have been, at least they've looked at it as being the scientific sweet spot or the creative sweet spot where you get really good ideas. In fact, a single minute of this N1 of sleep cycle can inspire insight. So what Dali would do, you may have heard about this, he would try and inspire himself for some of his creative activities, would go sit down in a quiet room, very quiet, would take a metal ball, put it there, and try to nod off. And as soon as the ball fell, he knew he was just in that moment, and he would jar him up, and he'd go beg and paid. Because it was this moment of trying to create that. Now, I'm not saying you do that. That seems much. But, you get the point, that there are ways in which you can ultimately use this. Quentin Tarantino, take him or leave him, but I think he's, written some pretty good, he's a thoughtful writer. He writes really well, and he does very well on some of his stuff here. But if you look at this, the idea for Django Unchained for him, he was listening to soundtracks of an old Italian composer, Mr. Morricone, and he said, something hit him, and he scribbled on the hotel stationery, the opening scene, word for word. And it was just this moment of, I'm just gonna, I won't say daydream, but listen to stuff, get inspired and write. And he didn't say, he says it in multiple interviews, I didn't change a word of it. And some of that, well, you can say leads to this, handwritten, and then leads to that. Not always that easy, but the point of the matter is it gets to the point of that these people seem to have ways in which they use this information. Now, you can show me the incentive and I'll show you the outcome. Fundamental. Do you believe that universities, education systems value thinking time? It's grossly undervalued. And it comes up all the time. It comes up in the editorial after editorial. Right? Thinking time is often undervalued, and it's rarely, if ever, quantified in academics. And that's the challenge, right? Because what are we incentivized to do? What are we incentivized to do in a university environment? Publish and get grants. And cycle, and repeat, right? So if you publish a lot, you go up. If you get grants, you go up. And obviously you can say impact is part, but there's a lot of, I know in my department, we can promote lots of people who publish a lot with no impact. Because the publishing itself seems to have an impact. The perception is that there's impact. And I think that's a bit of a flawed way to think about it, and I think we're not incentivizing the things we should be doing. And the incubation period is when you take that break, is where the magic happens. And I think that is really, a really important way to think about it. There's an effect associated with it, a psychological effect that says that you remember unfinished tasks. That's kind of the reason why you can argue, well, I take a break when I'm studying, because I'm figuring about a problem, and you walk away, you're doing something else, and somehow you're still working away on it subconsciously, and you come back and you're able to solve it. The other thing I think is fundamentally important, I've made a really, really personal effort to stay focused in the moment. So most of the stuff, I think, you wander through life, you wander through your day, and you miss the majority of stuff that's happening because you're just so focused on getting to what you have to do and what you need to do. And I think if you like the Stoics, you like Marcus Aurelius' quotes, the one that I like the most is the first one, which is be present. It's really hard to actually be present, not to look at your phone, not to do this. It's really hard, and you struggle, and I struggle with it. I don't know, I'm gonna ask you again. When you're climbing, you're pretty well present, right? I don't think you're probably daydreaming, just because of the consequences.
In between climbs, that's when I do most of my thinking.
Yeah.
The maps are very comfy, they're like a little bed.
Yeah.
You do
Right, but when but you're in the moment when you're, and you hear that with, with those who are playing instruments. You hear that with those who have art. Like I sketch, so these things are mine. Like I don't use those. Like when I sketch, I'm in the moment, and I'm really thinking. But I'm not really daydreaming much. I'm just in that moment. I took my daughter. This is now some years ago, to a pop singer, right, concert, and this was in Toronto. And I'm driving home. My wife says, "Oh, how many people were there?" I said, "I don't know, but there are 15,000 cell phones. There are 15,000 cell phones in attendance." And it is amazing to me how much of our lives are being recorded when we're actually there. And I grew up without cell phones, so I see both sides of it, and I can see the value of it. But I also see that this has become sort of the standard at most of these places. And I think it's really, really important, and I look at myself to do the same thing, is try to think of something simple and see it through you're seeing it for the first time. Anthony Bourdain did that beautifully. The late Anthony Bourdain did that beautifully. He would tell you a story about something, and he would say, "I'm looking at it as though I've seen it for the first time," and you'd feel it. So when you're talking about your research, you're talking about what you're doing, it's really, really important to do that. So we have tried at our own institution to do what we're calling the art of observation. So we take a small group of graduate students, and what we do is we take them to a place that we always go and say, "Okay, now wander around, take some photographs, and really pay attention, to the path you take 40 times in a week. Take a picture of that and come back to the group and tell us what you noticed this time that was a little bit different." Seems like a little silly exercise, but what was fundamental was somebody takes a picture and then the other group sees something completely different from what this individual saw, but then they had this really interesting discussion around the different perspectives. And I said, well, what if you looked at your data like that? What if you looked at other things you do like that? This is what you have to start doing, is really getting into thinking about, like, you're seeing it for the first time and engaging other people with that fresh set of eyes. I think the second thing that's really important is the network. High value networks are really important. In fact, you want to ultimately invest your time with the best possible people you can invest your time with. It seems very easy, and we often say, "No, I'm already with the best people." But I will tell you, when you take a critical look at your circle, right, you have to really think, okay, am I being challenged in the appropriate way? Is this the network that I think is going to help me? Let me ask you this: who's the fastest human on the planet? What comes to mind? Maybe I've got it wrong, but what comes to mind? Usain Bolt. Who's number 2, though? Perfect, right? But that's the reality, right? It is amazing to me how powerful it is and how much goes to the individual or individual teams that are one. The minute you get to number 1, you get all of the fundamental, I'd say, academic, if you're academic riches, if you're sports riches. I mean, he has his value is, if you take from 2 all the way to 10, is 10 times that. So what he gets is 10 times the rest of them combined. And that is a fundamental law that people talk about. And this has been published in journals, this Matthew effect. But basically, to those who have more will be given fundamental point. And why is that so important? And how do you apply that in this? It's about giving people opportunities. So look at this one. If you're writing grants, you know this. You're publishing a paper, you know this. You can take 2 individual people, identical in every other skill set. They write 2 beautiful grants, somewhat through chance. One scores 0.1 above one more, and one scores 0.1 below the cutoff at, let's say, the Canadian Institute of Health Research. Happens all the time. What happens to the individual early in their career that happened, you can almost say by chance, because there's 2 people, one got it, one didn't, they're both equal. What happens to the individual who actually got the early win? Look at what happens to their funding compared to the person who didn't. It is incredible how powerful opening a door and getting your first win is. In 1995, I did this study. By all intents and standards, it would be garbage compared to the quality of stuff I saw, but this was what I did as a resident, and we were looking at irrigation, and I looked at that, and at that point, I was a PGY2, second year orthopedic training resident, and I got my first award. I said, oh my gosh, I won something. And I got so excited by that, and that seemed to open doors, because other people, now suddenly people say, oh, maybe we can work with this fellow, we can do something and help him out. And that led to a bunch of other things. that opened the door and led to, and that one little study ultimately led through years to a large multinational trial many years later that led to a New England Journal paper. Now, would that have happened with or without that paper? I don't know, but I can tell you there was a very specific sequence of events that happened because of that paper, for sure. When you look at actual creativity and you look at impact and you look at papers that have followed scientists forward, it's incredible to say that most scientists peak 20 years into their career or by the age of 40. And if you look at Nobel laureates whom they've had their, they have the Nobel Prize and they'll have to have discussions, some of the critical things Nobel laureates go on about is we have to make sure our younger generation, the early career students who are building, don't get locked into a program of research because of the senior individual. We have to be able to explore part of their time because that's where they're going to have, they're the most opportunity for major insights are going to happen there because they're relatively undifferentiated. At least that's what they say. And it's an interesting. When you look at this, these are individuals who ultimately won Nobel Prizes out of a group of 40. This doesn't make any sense to you, but each of those boxes are meant to be different areas of research. The thing I was told is focus so narrowly in one area and be laser beam focused because then you'll become the world expert. True. But most Nobel laureates, from this case, they were involved in 3 to 5 different areas of research, which may seem counterintuitive, but when you see the transcripts of their interviews and why that's important, is because the fundamental thing about a truly innovative idea is to take something that's novel in this domain with something that's novel in this domain and combine it into something that's completely creative and new. And that combination through different disciplines is really fundamentally important. So if you never interact with individuals outside of your immediate circle of knowledge, you will miss that opportunity is what I'm gathering. And you know the thing that great scientists love to hang out with? Other great scientists. They just love it. They love hanging out with other high impact people. So your job, I think, is to get into the highest impact network that you possibly can. And looking at from what you have here, you have high impact networks here, lots of them. But I do think that's a really fundamental important. This is an individual, that individual I mentioned to you. So he has about 610. I don't know if anyone here even looks at, any of you students understand the idea of an H index? Yeah. So you know that citations are important because that helps you with understanding impact. There's all kinds of reasons why people say maybe that isn't the way to go, but it is the metric that lots of folks use. He's in medicine, he would be in the top 1% in the world of impact around H index and the citations. He has more than enough influence. But look at this. So I asked our library to go back and say, can you please do an analysis of the network of individuals before 1990, before the EBM revelation for him and the movement, and after 1990. You see the network grew very quickly for him. But each of those large circles is other individuals in his network that have an H index over 100. So he exemplifies a very, very high impact network, and he created many other high impact investigators by what would at least be a reasonable objective marker if you're looking across all the sciences. So there's a huge value in connecting with high impact in influencers and individuals. And this is kind of roughly what it looked like for him in his network. And so these are the people that he's been interacting with. So don't settle for the person in front of you. And these are individuals who talk about venture capitalists when they're coming to McMaster. They'll often come in and they'll say, you've got to find the right people. Right? And they may not be at McMaster. They're going to be here, but you've got to find and get in front of the best people you possibly can. Because when you do that, you're ultimately going to be able to make some big differences. And so that's been the movement in there. So third one, use your time wisely. So this is what I do. I'm not even arguing that whether it makes sense for you, but I'll share with you what recently in the last several years has been my personal vision statement. But I would tell you this, that when you actually take time, you find the right people you want to spend that time with, do something for yourself. Like, you spend a lot of time doing things for everybody else, but figure out what it is you truly do. And when you do something that truly is for yourself, as I've learned, you're actually doing the best thing you can for the people around you. You absolutely are. So you got to figure that out, and everyone's unique and everyone has their own belief. But I took this pledge in 2018. And I use the acronym THINK here, but that's just so I can remember it, quite frankly. And I've talked about it a lot. It's been things in pink have been, or light red, I don't know, they've been edited a bit, but try new things. Just go out there and try new things. Harness your creativity. I used to call that have fun, but I think it sounds more professional to say harness your creativity, so I've switched that a bit, but it's basically have fun. Try to have fun in your week. Invest in the 20% of the things that give you 80% of your purpose. And that may be a few things, but whatever it is, that becomes sort of, for me, it's the kind of a guiding principle of what I will do and what I won't. The reason I'm here, actually, is because of that guiding principle, right? Investing in things that I care about, building new networks, working with people that I ultimately want to build strong collaborations with. There's no other logical reason, right? Like to do stuff if you don't have a real purpose to do it, because you just go aimlessly around writing lots of papers and then you look back and say, well, what did I do all that for? Never feel failure, embrace it. Then I was in Mayo Clinic, and he said, well, maybe use that N for say no. Say no more often. Seems pretty obvious, but to do less is really hard, because those who do more get more. That's it. The more productive you are, the more people are going to throw stuff at you because you're the one getting it done. And then you just get more and more and more. Eventually you got to say no. Eventually you got to say no. And it's okay to start again. Like it's totally fine to reinvent yourself. Like this idea that I've gone too far in this path and I can't back out is nonsensical to me. So you can absolutely do that. As clearly as of the 2020 and onward, the kindness became a big fundamental issue. And this is actually our vice president of research at one of our hospitals in Hamilton. And he said, and I thought he wrote it pretty well. He was giving a farewell address, and I really, really thoughtful thought. But kindness is not a weakness. It's a strength. It builds trust. It fosters respect. It inspires loyalty. It transforms team into communities and elevates leadership to its highest form. So never think that being kind to people is being weak. But you got to be kind to yourself, folks. You got to look at yourself and say, I got to figure out what I want to do so I can ultimately make this work for myself. And here's the interesting thing. Successful scientists have highly integrated networks of enterprise, whereas their less successful colleagues tend to have fewer hobbies. This is a study that was done many, many years ago. 40 high potential scientists, all of which they thought were going to move on to Nobel Prize. 8 of them got a Nobel Prize. They went back over time and said, well, what made these individuals different? And they started exploring through qualitative work what the differences were. But it was fundamentally in your face that these individuals did things outside of their core work. And that's how they used their time. That's how they used their time, very wisely, I think, to do what they did. So I guarantee all of you have this, and if you don't, you'll look back in 10 years probably with that statement saying, I used to do this, but I just gave it up. I gave it up. I should get back to it. You made me think about this. I should get back to it. If you're doing something you love, keep doing it. It's okay. Whatever you end up doing, keep doing it. That's going to be part of the thing, I think, that'll keep you motivated into things you do. And if you've given it up, well, really think about why and how you can get back into it. This is a classic. It's one of the top albums of all time on many, many lists. So if you haven't heard her, you should probably go take a listen, regardless, just for the musicality of it. But she got a lot of trouble. She had a breakout. It was a blowout breakout album. And what happens when you have that? All the eyes are on you, and they said, do more, do more, do more, publish. She said, no. People in her world think I'm crazy because I keep taking big breaks between albums. And what do you think when they say, why do you take such big breaks? Why do you take 5 years? You could be making millions off of your albums. You can be cranking one out every year because she was so popular. What do you think her answer was? My best albums are born from a myriad of experiences. So if I'm not doing anything, if I haven't experienced anything, what am I going to talk about? And my argument is also the same way. When all I was doing in my residency was working and writing papers, I wasn't really experiencing much. I was just grinding through. So I wasn't you can argue, I was getting papers out, and that's fine, you're moving forward. But you're not really thinking about what's ultimately going to make an impact. So that took time to slow down. Time says, well, this is what you need. You need 7 hours of sleep. You need a hobby. Got to get out in nature. And you have to connect with people outside the home. If you're doing that, you're already off to a pretty good start. If you can keep that, that's pretty good. So this is Gordon Guyatt. Him and I will chat all the time. and we'll go out and we'll catch up and we'll do all kinds of things. And we've written some work together that actually says, hey, if you're outside walking, you actually have more creative thinking. And in fact, if you're just walking, like walking is better than sitting. And if you're walking outdoors, it seems to be a little bit better than walking indoors because you can just see the incremental benefits. But there's more than enough data. We took our recent three of our PhDs. These are certain scientist PhDs. They were just finishing up. So it's okay. Pick a place you're going to go. You have to invest in it. You have to be invested. It's not a free trip. And pick something hard. And if we fail, we fail, but at least we give it a go. And I think that's important. And I said, well, when we finished it, we have all these discussions, and they had their personal diaries, and they were writing things about life and whatnot. But one of them just said, I'd encourage all students to consider taking time out of your day, week, or even year to invest time, energy, and money into something that makes you uncomfortable. I think that was the goal, right? That was the part of it that got of it. So create time, build networks, and when you have all that, use it really, really wisely. That's the message I would give to my younger self, absolutely, in doing that. And I'll give it a caveat here as I finish off with, there's some three, I guess, simple truths in my mind. And I've recently come across some stuff by Peter Singer, but he's, again, considered at least by many as being one of the more thoughtful of the bioethicists that are currently living. And it is fundamental, right? Like, so I travel a bit. I get the privilege of traveling, and I'll go, let's say, I was in Japan recently, I'll go there, and then I'll say, we got to do stuff. This is great. Like, you're right in front of me. Your issues are exactly the issues I see when I go back to McMaster. And what happens the minute you have distance, it just, even though you have the exact same issues, the person right in front of me gets all the attention. And we forget about that, right? So when you're thinking about building these networks and you're building things, know that you're not alone. I would guarantee that there's a group of students exactly like you in every part of the world who have the exact same, future, beliefs, dreams, hopes, and fears and challenges. And the thing is, how do we ultimately, as a department, as this institute, as you think about the center, how do you also reach out? I think there's a lot of international support here too, but how do we do more of that and move that forward? Because ultimately, what happens is, okay, I'm right here, and we're all in different parts of that. Everything I have is forward. I got to take some risk. If I don't do it, I'm going to have more and more regret. And the only thing stopping me is this belief that, I got lots of time. I got lots of time. Second point. For me, people say, well, what's this urgency? Well, there's no urgency, it's just age. Age is your alarm clock. Every tick, 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 that's it. You just keep moving along. So you think every second I'm not doing something I want, I'm going to have regret. So it's a matter of when, and you can be 15, you can be 18, or you can be 75. It doesn't matter. Like, you might have slightly different perspective on that time, but it's still the same thing. And I think the thing that really has been important is impact does not happen overnight. You can be very productive. Like, you can be productive in a month. You can start cranking out papers. I'm not saying it's happening here, but I can tell you that there's definitely, you can start moving. When you get into a very highly productive group, that's, I'd say, a paper mill, you can write papers. But the problem is most people, this happens with our surgeons. So every year we have about 150 surgeons, and I'll meet with many of them, especially those who are in their first year. They'll meet with me as the chair, and I'll say, well, tell me what you want to do next year. You know what they'll do? They'll give me a list like this. I'll say, oh my gosh, that sounds amazing. In my head, I'm thinking, there's no way you can do any of this, right? Because it's just too much. But you will grossly overestimate what you're going to do in 1 year. But you know what most people forget? They grossly underestimate what they're going to do in the long term if they stick at it. Because that's the fundamental, the shifter. Because 1 in 10 of us say, okay, I'm going to do it. 1 in 100, well, I'll actually do it. And then when you have that ability to say, I'm going to stick at this, like I'm going to really stick at this, that is the absolute rare. And it shouldn't be that rare, but it is. So it doesn't take much to keep at it. And the thing is, even if you're, quote, failing, or you feel failing, you're not. You're actually learning every time you push this forward. I don't push books too much, but if you haven't read The Alchemist, it's such an easy book to read. You can read it in 2 hours. I highly recommend you read it. You'll think it's a children's book. I did. And then it just hammers you like a week later. Like, oh my gosh. And then you just start thinking about it. And then it just becomes on your mind the whole time for about a while. And then you read it again and you get something else out of it. But something he says, Paulo Coelho says, is when you take the first step, people, forget the universe part of it, but people Doors will open for you. When someone sees that you're trying, people around you will notice that. They'll see the enthusiasm. They'll see the infectiousness of your interest, and that will engage them and say, we should help this person. We should do something. And if you do one thing, just one thing from any of this I've been going on about, just one thing, make yourself a contract. Call it whatever you want, right? I'm going to spend, 15 minutes, whatever that is. I'm going to spend something doing something I'm going to spend time even thinking about it. Just doing something, that will already be an action. And that action may lead you to something else, and it may not. But I actually feel that you can actually, when you get excited about something, and you've seen it, someone comes in, excited, the whole team gets excited because it's infectious, right? So you want to make sure that whatever you're doing, that you're really enjoying it, right? And you're doing your best to try to find that path. Now you could say, this guy's been babbling on, and it came in, I may be wrong, but you may look back in a few years, maybe for some of you many years, and say, holy, he was exactly right. It's exactly what I should have been focusing on. Because I've been there. Like, I've seen it. I was every one of you. I was a student. I was in a lab. I was doing everything you've done. And I was trying to think about the future. And I had lots of advice. I followed a bunch of it, a lot of dead ends, and a lot of great opportunities. But at the end of the day, the late Ozzy Osbourne, I had to sketch him because he just passed. And if you grew up in Hamilton, Ontario, that's what it was. You were listening to heavy metal and rock and roll in the 1980s. So it's an interesting point, right? The thing about life is that by the time you're older and you begin to understand, you die. A bit macabre, but the truth of the matter is age is your alarm clock, friends, and think about that fundamentally. So to the students, to the faculty, let creativity become your habit, not your hope. Thanks so much, folks.
I think we have time for some questions.
Oh, sure, yeah.
Thank you for the inspiring talk. I was wondering, because right now we live in a world you can just Google and then probably find a solution. So how do you balance a creativity, or you just rely on the internet? Do you have any suggestion about those?
The truth is, the internet's one thing, but forget Google. I would say that we're into all the chatbots, we're into all the AI, right? We're into all of that, and I think, in many ways, I think it can be very powerful to use that. All of our students use it, but I think it fundamentally gets back to the idea. So you can question a chatbot and get answers, but I do think you need human interaction. Diverse human interaction. So what we've been trying to do from the point of view of teaching and mentorship is making sure that our graduate students and our students who are working with undergraduate students have a really diverse group of individuals with whom they can check ideas. It's not just one individual. And then they can stress test all these different things. So that's been our big thing. We're really trying to connect them with one thing. I'm saying what I said, which is getting them an early win in their short time with us, and getting them connected to a very highly quality network.
I have one. So one of the things you mentioned is to do less, focus on quality, and it seems, or actually it is, the entire scientific enterprise is engineered to incentivize people to do exactly the opposite.
Yes.
Do you have any suggestions on any suggestions at all as to how this can be reversed?
Well, I think the first thing is you have to talk about it and raise it. I give versions of this to lots of groups, and it's just varying degrees of interest. It just depends who you are. But I think getting in front and talking about it and having an open discussion, I don't think it's really that unintuitive that our education system really is doing the opposite of what's purporting it's supposed to be doing. It's not making us more creative. It's not happening. And people have been talking about it for decades, right? And there's just paper after paper that says the same thing on and on again. But the biggest fundamental challenge is, what does it mean to be creative? What is creativity? if you talk to some people who are very successful, they'll just say, yeah, I am creative. Like, well, what do you define that? He goes, well, you know, and then that's where the whole debate happens. But I think we should talk about it. And I think our leaders at the hospitals and at the university should talk about it. Yeah.
Are there any more questions?
Maybe I have a question. I mean, in the last couple of years, we had the statements all around, like the science is settled. There is consensus of 99% on this kind of things, right? So I think that's one of the reasons that the creativity goes down. And some people which are out of this box, they get discriminated.
100%.
So that's horrible development, right?
We're having this discussion. Try to get a novel idea funded at CHR, like really novel. It's going to be reviewed by scientists who are going to say, well, this doesn't make any sense because this is not the theory that we go forward. If you look at most of the absolute, complete global disruptors, they were pariahs actually in their time. Now, I'm not saying I have no desire to be hated by the community just because I have an idea, but I also have a desire to say, how do we get this out there and how do we get people like-minded to start thinking about it? But you're right. And that's why I do think universities broadly have a real opportunity, because this work, some of this truly trailblazing work is not going to get, game-changing work is not going to get funded through traditional pathways, I don't think. So we have to find that money and support these individuals. And we never look at someone's resume and say, my goodness, this person tried 15 things and failed at every one of them. My goodness, promote him or her, right? So like, if you had a failure resume, and it was like, whoa, this person's trying a lot of stuff, we should be like thankful that someone's trying a lot of stuff. But that doesn't happen, right? We only see the stuff that succeeded, and that's unfortunate. But I don't know. I mean, these are things that we're having discussions. But I know intuitively we understand that there's a problem with the system. But how do you fundamentally change it? And I think you start by talking about it.
Yeah, so you mentioned that great breakthroughs come from those who are unreasonable. For a student, especially one with limited skill sets, what would you recommend, or what would that look like to be unreasonable as a beginner?
Well, sorry, just to be clear, I didn't say that. Rick Rubin said that. But,
right.
But that being said, I do believe it. Like, I do think, to the point we've just said, right? When you're in a lab, and I don't want to take up, like, I know there's a venture. Okay, so, let me ask for the students. Okay, so you've been working on great projects. During your time, when you're working on a project, I'm assuming you were, assigned a project and you've learned about it and you've taken it on. Did you ever have insights like, oh, I wonder about this, or I wonder, maybe we should do something completely different? Or you've any daydream about anything that would be completely off of what you were doing? Anyone? Show of hands. Yeah, okay, so there's a bunch of you. So what did you do with it? What did you do with that insight? Kept it to yourself? Buried it down low? No, but did you raise it? Did you say, oh, we should do this and we should try it? Or did you raise it in the group? And I guess my point, I'm not asking for an answer, it's a rhetorical somewhat, but the point is, like, we have a tendency to think that our ideas when you're earlier in your career, this doesn't make sense, because like I don't have enough knowledge in this area. But also keep in mind is because you don't have knowledge, you may see things that is like someone like me who maybe worked on something for a long time doesn't see. So the biggest, I think, disservice you can do in a team environment is not to share that. Worst case scenario is, oh yeah, you learned something. Oh, maybe that doesn't make a ton of sense, and here's why. But you might actually open up a whole area that could be really interesting. So I think we often think you have to have immediate domain expertise, but I'm telling you, like by 29, like they say at the postdoc level, if you look at a lot of the Nobel laureates, not to say that everyone's aspiring to be a Nobel laureate, wouldn't it be nice? But the truth is, the insights have come very early in their career. They weren't late, they were very early, and it took many, many years to explore and develop them and get them proven. So you're really at your sweet spot for many of you, to be thinking as creatively as you possibly can. So what are you doing, I guess, in my mind, to cultivate that? That's all. I don't know if that helped.
Any more questions? Hearing none. Dr. Bender, thank you so much.
Oh, thank you.
Thanks so much. Yeah. So I have just a couple of announcements here. Pull up the original slide deck. So once again, sincere thanks to our sponsors for making this entire event possible. And a reminder that what's next is our poster session with food and drinks. We have a number of poster judges who still need to pick up their poster judging forms at the front desk, so please see Mira for that. And we have a dinner and awards immediately after the poster session, so please do enjoy this. So once again, thanks again to the sponsors, and thanks again to Mira and Jessica, Sharda and Dipali for all the work behind the scenes, not only for this event, but also really throughout the year. And thanks once again to the students. Congratulations on really an amazing job. And thanks again to the mentors. And please enjoy the rest of the event. Thanks a lot. Okay, great. Okay. They know about this, right? Okay. So could all the students please gather at the front for a group photo, please? All the students. Thanks.